students? Am I audible, visible? Hi students, am I audible? Am I visible? Hi guys, good evening. Welcome to Unacademy NEET English. I hope you are doing great. I hope you all are doing great. Am I audible and visible to you all? Yes, please. Am I audible and visible to you all? Good evening, good evening, good evening. Welcome to the session, guys. So today we are going to discuss neural control and coordination. You know that in game of NEET, when I was taking this chapter earlier, so something happened and because of some technical glitch, I was not able to continue the session. So today we are going to discuss it. I thought that I'll put a recorded video, but honestly, I didn't get the time for that. Okay, so it's better to discuss this chapter, right? If you guys are having any doubt, you people can ask me. I'll resolve that doubts and we will finish our chapter. And yes, for the enzymes, definitely for that you will get the recorded video, because that's not a very lengthy topic. Okay, that's not a very lengthy topic. Okay, so today let's discuss the neural control and coordination. Okay, so if you are excited, do let me know in the chat section. If you guys are excited, do let me know in the chat section that yes ma'am, we are ready to finish this chapter. We are ready to finish this chapter, because if you are not, then I will also end the class. I'll sit, because it's too cold here, right? It's too cold here and it's really challenging to take the marathons. Okay, chaliye. So let's start, guys, and I hope you people are aware that our Avengers batch is going very fine, our Avengers fast track batch. And in this batch, guys, the plan is clear. You know that we have told you already that in the month of March, till 31st March, we are going to finish your syllabus. Along with that, every week you people are going to get tests. Right, in the month of April, students, we will have 30 full syllabus mock tests. So if you join this batch, you are going to get the test series, you are going to get the proper content, proper schedule, proper revision, everything. So without delay, be the part of this batch. We are discussing cell cycle and cell division here. Soon, after biomolecules, we will move to the genetics part. So this is what I'm planning for the biology, that in the month of, yes, that till the month of January, we have to, that's our target, we have to finish, right? We have to finish the most important chapters, right? We have to finish the most important chapters. That is our target. Okay, so neural control and coordination, right? Now let's discuss it. So do not forget to join the batch. Okay, chalo. So now let's start it. The neural control and coordination. This is the chapter that we are going to discuss. So the simple word is written here, that is coordination, right? There is a very simple word that is written here, and that word is coordination. Yes, that word is coordination. What do you understand by this word? Coordination. 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 Yes. What is the meaning of this particular word? Coordination. What do you understand by? Right? What do you understand by this word coordination? See, it's very simple, right? Because it is the human physiology, right? It is the human physiology. You know that we are multicellular organisms. What are we? We are multicellular organisms. Yes, students, what are we? We are multicellular organisms. So in our body, we have so many cells, right? We have so many cells. There are so many cells that are making our body. So when you talk about the multicellular organism, you know that cells, group of cells, they form tissue. Then that tissues, they form organ. That organ, they form organ system. And different organ systems altogether, they form organism. Different systems altogether, they form what? They form organism. What do they form? They form organism. They form a living body, right, bache? They form a what? They organism. They form a living body. You know it very well, right? So obviously, if in an organism so many cells are there, right, it's not like that they'll work individually. No, of course, not possible. There should be a coordination. There should be a coordination. They need to work together, right? Then only our system can work properly, right? Then only the things will be in a sync. Then only the system will be like a harmony will be there. That's what we can say, right? That is what we can say. So they need to work together. So when it comes to the coordination, right, students, in our body, we have the nervous system for that, right? Neural system, neural control and coordination, right? Neural control, neural coordination is there, and we are also having the endocrine system. It's not the nervous system only, the endocrine system is also there, right? The endocrine system is also there, not just the nervous system. So when I'm saying nervous system, when I'm talking about the nervous system, of course, we are talking about the neurons, we are talking about the neuroglial cells, right? Their function, the way they coordinate, right? That's what we discuss here. When we are talking about the nervous system, you are talking about what? You are considering the brain, you are considering the spinal cord, you are considering, basically you are considering the neurons, neuroglial cells, brain, spinal cord, different nerves. That's what we are considering. That is what we are considering. And when it is the endocrine system, you are talking about the chemical messengers here, right? What do we discuss here? We talk about the chemical messengers here, right? Information molecules, chemical messengers, hormones which are synthesized in a minute amount, right? Which are synthesized in a minute amount. That is what we discuss here, right, bache? That is what we discuss here, right, bache? So neural system and the endocrine system, they jointly coordinate, they integrate all the activity of the organism together. So when it comes to the coordination, there should be a system that is taking care of it. There should be a system that is ensuring that everything is working properly, right? Everything is working properly in a sync. Yes or no? Yes or no? Right? That should be the point here. Definitely that should be the point. Okay. So now, when it is the nervous system, right? Nervous system, endocrine system. That's what I just said. What do we have? Nervous system and endocrine system, right? So they are making sure that body is working in a coordinated manner. That is making sure that the body is working in a coordinated manner. Is that clear? Right? Right? So you know that we have dedicated system for digestion, for excretion, for reproduction, for respiration. We have the systems, na? We have the systems, na? So same way for coordination, these are the systems. That's what you need to remember, and that is very important as well. And it's not like that even in only in the multicellular organisms you will see a kind of coordination, or only multicellular organisms, they respond to the stimulus they receive. No, even the unicellular organisms, even they do respond, right? In their case, they say chemotexis, these kind of things are there. Okay, these kind of things are there. So let's discuss this part. So you people can see here in the NCERT that the functions of the organ or organ system in our body, they must be coordinated to maintain homeostasis. So please tell me, what is the meaning of homeostasis? Anybody in the class? What is the meaning of homeostasis? Yes, what is the meaning of homeostasis? Yes, students, please tell me. What is the meaning of homeostasis? What is homeostasis? Maintenance of internal environment. Yes. So when we are saying homeostasis, basically it means the maintenance of constant internal condition. What is it? It is the maintenance of constant internal condition. What is it, bache? It is the maintenance of constant internal condition. The internal conditions should be constant. They are not fluctuating, right? The internal system is stable. That's what I can say. That's what I can say. Right? Ashish is writing that to maintain body temperature relative to the environment. Are you sure about it, Ashish? Right? So in simple words, if I have to define, so it can simply say that to maintain the stable conditions in our body, to maintain the stable conditions in our body, that's what we can say. Right? That's what we can say. So when it comes to the homeostasis, right, to maintain the constant internal condition, you know that in our body, right? In our body, thermoregulation, osmoregulation, both the things are there, right? Thermoregulation, osmoregulation, that is salt, and that is to maintain salt and water balance. Thermoregulation, you're talking about the heat. So these kind of things, these kind of regulations are ensuring that our body, right, our body is having a stable environment. For an example, when you talk about the poikilotherms, students, what are poikilotherms? Anybody here in the class? What is the meaning of poikilotherms? What are poikilotherms? Yes? What are poikilotherms? Please tell me. What are poikilotherms? Please tell me. Yes, please. Cold-blooded animals, very good. Cold-blooded animals, very good. Yes, they are cold-blooded animals, right? Like fish, amphibian, reptiles, they are what? They are the cold-blooded animals. So in their case, their internal conditions are not constant. It changes with the environment. It changes with the environment. Environment is affecting it. It is changing. The internal conditions here, they are changing with the environment when you talk about the poikilotherms. So they need to go for the winter sleep, summer sleep, right? When, you know, the temperature, like when they cannot bear that particular temperature, they just move. There are different ways, right? There are different ways to, there are different ways of their survival, but yes, it is the thing in their case. Right, but on the other hand, when you are talking about the homeostasis, right, the constant internal condition will be there. Take the example of human beings. Just we say that we have our temperature is 30, what is it? 98.4 degree Fahrenheit, right? Right, that it is constant when you talk about the temperature, right? It is constant. So obviously it is energetically very expensive, and to maintain the homeostasis, right, the kidneys are there, they are helping in the osmoregulation, thermoregulation is required. So basically even that is also the thing, that if we want the homeostasis, right, we want the stable internal conditions, the body organs should work together in a coordination. So when you are saying coordination, means all the body organs, they are working together in such a way that they are complementing each other. Okay, now the word is integration also. So when you are saying integration, bache, जैसे coordination, okay, fine, ma'am. Body organs are working together in such a way that they are complementing each other. But what about the integration? Integration will make these two or more organs, when it is integration, to integrate. So basically two or more organs, they are working together and they function in harmony. That's the point. They function in harmony. That's the point. Is that clear? So coordination is a process through which two or more organs, they interact, they complement the function of one another. Right? Like when you are doing physical exercise, our body will demand more energy, right? Why? Because increased muscular activity is there. Increased muscular activity is there. Yes or no? Yes or no? So supply of oxygen is also increased, right? So that is how body organs are complementing each other, right? So obviously if there is high oxygen need, definitely the rate of respiration, the heartbeat, the blood flow, it should also be high. Right, bache? So when there is physical exercise is stopped, the activity of nerve, lung, heart and kidney, it will return to their normal conditions. It will come back to its normal condition. So the function of muscle, lungs, heart, blood, vessels, kidney and other organs are coordinated while performing physical exercises. Right, bache? So neural system and the endocrine system, right? Your hormones, they work together. Right? Okay. Okay. Chaliye. So next thing here is, see, this is what they have given, that when you talk about the neural system, it provides an organized network of point-to-point connection for a quick coordination. Yes, bache. When you are talking about the nervous system, you are talking about the neurons, point-to-point coordination is there. The response will be quick. On the other hand, when it is the endocrine system, when you are talking about the hormones, take one example. Take the example of your pituitary gland. Pituitary gland is releasing one hormone. It will act on its target organ. Then the further, right? Further steps will be taken. So obviously that is not very quick. That is slow. That is slow. So when you are talking about the neural system, point-to-point coordination, point-to-point connections, right? Point-to-point connection is there. Point-to-point connection is there. Point-to-point coordination is there. Is that clear? Is that clear? And here in endocrine system, you have the hormones, and what do we call them? They are the chemical messengers which are required. We have already completed this chapter now. It is the chemical, these are the chemical messengers that are required in minute quantity. They are not some nutrients. They are not some nutrients. And tell me, bache, tell me one thing. Chemical messengers, are they intracellular messengers? Hormones, are they intracellular or intercellular? Hormones, what are they? Are they intracellular or intercellular? Are they intracellular or intercellular messengers? Please tell me. Please tell me. Are they intercellular or intracellular messengers? Hormones. Hormones. They are intercellular, in between cells. So they are intercellular messengers. Intercellular messengers. Yes. They are intercellular. Aparna, read it carefully. Yes, read it carefully. So what are they? Intracellular or intercellular? What are they? Inter or intra? Again, are you sure it's inter? Is it inter or intra? Is it inter or intra? I don't know. You have to tell me. Is it inter or intra? Be sure about it. Intercellular messenger required in trace amount. Okay. Okay. Chaliye. So let's discuss about this neural system. Let's discuss about this neural system. And what are they saying? That neural system of all animal is composed of highly specialized cell called neurons. And these neurons can detect, receive and transmit different kind of stimuli. Okay, bache? Okay, bache? Chalo. Okay, chalo, let's discuss it. So, before starting it, let's relate the things with animal kingdom. We talk about the porifera, and poriferans, they are having sponges. We talk about the porifera, and poriferans, they are having the sponges, right? Like porifera, pore-bearing organisms. What do you think? What do you think? Yes, do we have the nervous system in the case of poriferans? Do we have the nervous system in the case of poriferans? Yes. Do we have nervous system in the case of poriferans? You're talking about the porifera. You're talking about the sponges. You are talking about the porifera. You are talking about the sponges. So sponges are also multicellular, right? So many cells are there, but they don't have tissue level of organization. They are multicellular, they don't have tissue level of organization, but they have many cells. So what are you talking about? You are talking about sponges, right? Sponges. So in the case of sponges, there is no nervous system, bache, right? Keep it in your mind. When you talk about the animal kingdom, right? In the sponges, there is no nervous system, but still they respond to stimulus. Yes, stimuli. Right, chemicals are there for that. They respond to their external stimuli, chemicals through the chemicals, right? That chemotaxis is there, but nervous system, yeah, neurons are not there in the case of porifera, right? So now when you talk about the next phylum, Cnidaria, that is your Ctenophora. Ctenophora, yeah, Coelenterata. It is your Coelenterata, right? Ctenophora or, sorry, Cnidaria or Coelenterata. So we have the example of Hydra there, if you remember. Right, we have the example of Hydra, remember? Polyp, Hydra, yes. We have the example of Hydra, yes. So in the case of Hydra, we discussed now there is a diffuse network. There is a diffused network of nerve cells. There is a diffused network of nerve cells. Yes or no? There is a diffused network of nerve cells there. So obviously you are not going to see a kind of brain here, but again nerve cells are present here, right? Nerve cells are present here. So in the phylum Cnidaria, right, for the very first time we will see that nervous system, a diffuse network of nerve cells will be there, right? A diffuse network of nerve cell will be there. Understood? Understood? Sure? Sure? Yes? Any doubt? So neurons are not there in your Cnidaria. Sorry, neurons are not there in your porifera, right? But they have the cells, right? They have the cells that will respond to that. So in the hydra you will see, right, for the first time that there will be, what will be there? Diffuse network of nerve cells. Understood? Diffuse network of nerve cells. So obviously when we move from one phylum to another, we will see that things will move from simplicity to complexity, right? Things will move from simplicity to complexity, right, bache? With the time we will also see, because here you have the neurons in hydra, but you are not going to see the brain. With the time we will see that even brain will develop, not directly, right? Slowly that thing will be there. Just for an example, there is a word, cephalization. There is a word, cephalization. Cephal word is associated with your head. So cephalization means head formation. And we will see that organisms, right? In the case of organisms, we will see that when we study the things from porifera to chordata, we see that how the advancement will come, right? How, from their simple body, the things will become complex. So if I'm talking about the cephalization, if you remember planaria, yes, if you remember planaria, do you remember planaria? It's a platyhelminth. Planaria, it's a flatworm, it's a platyhelminth, also known as Digasia. It is having great power of regeneration. So in this case, you see ladder-like nervous system. What will you see? Ladder-like nervous system, right? Some degree of cephalization, some degree of head formation will be there. And then let's take the example of annelids. Let's take the example of annelids, right? So we discuss about the, yes, right, you know that we have dorsal hollow nerve cord. Do you remember? Yes, Arun, we have dorsal hollow nerve cord. So in the case of, we are chordates, our nerve cord is on the dorsal side. We are chordates, our nerve cord is on the dorsal side, and it is hollow. It is hollow. It is hollow, single nerve cord. And when you talk about the non-chordates, in their case, if there is any nerve cord, so it will be on the ventral side. It will be on the ventral side. It will be double. It will be solid, right? It's not hollow. So obviously with, like, when we move from porifera to chordata, we will see that how things will move from simplicity to complexity, and we will see the changes in the nervous system as well. We will see the things that how the things how the further complexity will come. Okay, so nerve cords, the ganglias, that's what we have to see, right, bache? Right, bache? That's what we have to see, right? Yes? Yes? है ना? That is what we have to see. So not just the nerve cord, ventral side pe solid double nerve cord is there, right? Not just the nerve cord, you also talk about the ganglia that is composed of nerve cells, that are aggregates of nerve cells. That's what we see. So if you talk about the, I'm not talking about the chordates only, not just specifying the non-chordates only. If I talk about the invertebrates, when I'm using the word invertebrates, means they do not have vertebral column. And even in phylum chordata, when you talk about the urochordates, right, you talk about the cephalochordates, even they do not have the vertebral column. I believe this is clear, right? Even they do not have the vertebral column. Even they do not have the vertebral column. So in their case also, bache, right, overall in invertebrates, you will see that octopus is very intelligent animal. Octopus is very intelligent animal. So octopus is having highly developed brain. In invertebrates, octopus, your devilfish, that's a mollusca, is having highly developed brain. Understood? They have highly developed brain. Okay? 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 So keep it in your mind. Done? Okay? Chalo. So now let's discuss about this neural system, nervous system. Obviously, we need to discuss the neurons first, right? What are neurons? What are nerves? Right, bache, how are they different? We should discuss it, we will. But firstly, let's talk about the nervous tissue, right? Let's talk about the nervous tissue, that what exactly do we have here? Yes, what do you know about the nervous tissue? What do you know about the nervous tissue? Please tell me, what do you know about the nervous tissue? Yes, please. Yes, please. Nervous tissue. Bache, your nervous tissue, it is ectodermal in origin. What is the meaning of being ectodermal in origin? Ectoderm is a germ layer. So it is made up of, right? It is made up of that germ layer, right? It is ectodermal in origin. It is made up of germ layer. So it will contain neurons, also known as nerve cells. And you know that in the human body, it is the longest cell. So neurons, nerve fibers. Yes, please write down neurons, nerve fibers, then the bundles of nerve fibers forming nerves. I will simply write nerves, right? Your neuroglial cells, right? Yes, neurons, nerve fibers, nerves, your neuroglial cells, your other cells are also there, like some epithelial cells, you can call them as appendymal cells. You have neurosecretory cells. So they all are making your nervous system. So in the nervous system, in the neural tissue, in the nervous system, in the nervous tissue, you do not have just neurons. You do not have these nerve cells. You have nerve fibers, you have nerves, right? You talk about the neurosecretory cells, you talk about the appendymal cells, you talk about the neuroglial cells. Is that clear? Is that clear? So firstly, let's understand what is a neuron. Yes, and you tell me, what do you know about a neuron? Let's talk about the neurons. And that's my favorite diagram. I hope you know that. I know you will be like, madam, we have already completed it in our other lecture, but that's okay. Let's discuss it and revise it, and you will also get extra points. That's how we draw it, bache. That's how we draw it. Yes or no? Something like that. That's the neuron. That's the neuron, and that is the nerve cell. We also call it as nerve cell, right? Chalo. So this part here is cell body or cyton or soma or perikaryon. Yes or no? Cell body, cyton, soma, perikaryon. This is what we have. And from the cell body, these processes are arising. Like these branches like process, they are dendrites, right? They are dendrites. And here the cylindrical structure, this part is axon. This part is axon, right, bache? Now when you look at this, it is your Schwann cells. Matlab, this one actually, right? Or in this diagram, it is not that clearly visible. This one actually, and this layer is made up of myelin. It is the myelin layer. right? It is the myelin layer. We'll discuss it in detail. So now when you see the gap, just say this, imagine a layer is covering a part of it, right? And you have a cell here, Schwann cells, right? Schwann cell. And see, here there are the gaps where myelin sheath is absent. We call it as nodes of Renvir, right? That's what we call it, nodes of Renvir. So these are the axon terminals, or we also call it as terminal arborization or axon endings. It is also named as terminal arborizations or axon terminals or axon endings. And bache, here this part, right, some structure like this is there, and that is known as axon hillock. What do we call it? Axon hillock, right? What do we call it? Axon hillock, right bache? Okay, so that's what we have. Okay, so cell body, from that cell body, these processes, neurites, which are dendrites or axon. This part is axon hillock, very sensitive part of neuron, right? This is the myelin sheath, it is covering it. The gaps in between where myelin sheath is not present, it is nodes of Renvir. These are the terminal ending. This is the basic structure. Now we have to discuss it. Okay, this is a kind of basic structure. Now we have to discuss it in detail. Okay, so this is the neuron, and let me, this is the neuron, and let me tell you, students, it is the basic functional unit of nervous system. It is the basic functional unit of nervous system. Yes or no? It is the basic functional unit of nervous system. What? Neuron. Okay. What? Neuron. Okay. Chaliye, let's discuss it. Any doubt? Any doubt? The diagram part clear? The diagram part clear? So neurons, what are they? They are the, yes, neurons, what are they? They are the basic functional unit of nervous system. They are the basic functional unit of nervous system. Okay. Okay. Yes. Okay, chalo, let's go. So after that, one by one we will discuss about that fibers and all, nerve fibers and all. So it is the basic functional unit or structural unit as well. Structural unit as well. So when you talk about the structure of neuron, right? So it is having it is the basic structural and functional unit of nervous system. Right, functional unit of nervous system, right bache? Now here the point is, it is having two things. One is your cell body, and right here you have two things. One is your cell body and another is your cell processes. One is your cell body and another is this processes, cell body. As you know, we call it as soma, cyton, perikaryon. Right, it is known as soma, cyton or perikaryon. And when you are talking about the cell processes here, your cell process here, we also use the word, and you can call them as process of neurons also. Right, you can also write it like this. Number one, cell body. Number two, number one, cell body. Number two, process of neurons or cell process, right? So you and yes, neurites. The another word is neurites, and that is your dendron and axon. So dendron is singular, right? Dendrites are plural. So dendrons and axon. That's how you can remember. Okay, that's how you can remember, right? Right? Okay. So cell body, soma, cyton, perikaryon, cell processes, right, also known as neurites. You have the dendrons, you have the axon. Understood? Yes, understood? Yes. ठीक है. So now when you talk about the cell body, obviously cell body, nucleus is present there. Nucleus is present, it is uninucleated, right? Nucleus is present, it is uninucleated, or you can say that except centriole, all other cell organelles are present here. Except centriole, all other cell organelles are present here. So centriole, you know about the centriole, right? When we talk about the centriole, you know that it's a non-membrane bound organelle. Yes, it's a non-membrane bound organelle. It's a non-membrane bound organelle. You will see it in the animal cells, or right, you will see it in the animal cells and in some lower plants also, but in the animal cells they are present, right? But they are absent in the higher plants. Okay, and you know that the centriole duplicate to form centrosome, and that will form spindle apparatus. Right, that will help in division. So obviously centriole is not there. So in the case of neurons, we know that, right, they will be there in the G0 phase, right? Their G0 phase is kind of permanent, right? This quiescent stage or cell arrest stage is kind of permanent. Matlab only at the time of like when zygote is growing, right? At that time when zygote is developing into a complete organism, only that time you will see the nerve cells are present. Later on, they do not divide. Right, later on we say that they do not divide. They will only form at the time of birth only. So we can say that they are in the cell arrest phase, and that cell arrest phase is kind of permanent, right? It is permanent. And in their case, in the case of neurons, the metabolism is going on, right? The metabolism is going on, the chemical reactions are going on, but it's just that they are not dividing. So now you know the centriole is not there, that's why. Okay, so except centriole, all other cell organelles are present, plus here in the cell body, in the cytoplasm there, which is also known as neuroplasm, what will you see? What will you see? You will see Nissle's granule and neurofibrils are present. Neurofibrils are present. So now when you talk about the Nissle's granules, now when you talk about these Nissle's granules, so students, they are the site of protein synthesis. They are the site of protein synthesis. What are they? They are the site of protein synthesis, and they are what? They are the aggregates or masses of. They are the aggregates or masses of ribosome and endoplasmic reticulum. Of ribosome and endoplasmic reticulum. That's why it is the site of active protein synthesis. So Nissle's granules, bache, they are the site of your center of protein synthesis, right, bache? So it is the your endoplasmic reticulum and ribosome. Its aggregates are basically the Nissle's granule. They are the masses of ribosomes and your endoplasmic reticulum. Okay. Okay, so remember this thing, this point is important. This point is important. In the NCERT also it is given, but not properly. When you talk about the Nissle's granules, they are present in cell body and dendrites. They are present in cell body and dendrites, but absent in axon. Okay, but absent in axon. But where are they absent? They are absent in axon. So they are the aggregates of what? Aggregates of ribosome and endoplasmic reticulum. So it is present in cell body and dendrites, but it is absent in axon. This information is important, right? This information is important. Okay. Now the next thing is neurofibrils. Now the next thing is neurofibrils. So these neurofibrils, what are they? It's the cytoskeletal element, of course. It is the cytoskeletal element, of course. What's that? It is the cytoskeletal element, of course. What? Your neurofibrils. So they are basically meant for what? Meant for the movement of things, yeah, internal conduction in cyton. internal conduction, because cytoskeletal elements, they are required within a cell. They provide the shape, right? They provide the shape, they maintain the shape, they help in the movement of things from one part of the cell to another, right? So you can say internal conduction in cyton, ya fir movement of things within the cyton. That's what you can say. That is your neurofibrils, right? What are they? They are your neurofibrils. Is that clear? Is that clear? So these are the small proteins, of course, right? These are what? These are the cytoskeletal element, means they are the proteins, and it is actually the intermediate filaments. What are they? What are they? They are the intermediate filaments. What are they? They are the intermediate filaments. Clear, bache? Clear, bache? So now the next thing is dendron. Dendron. So that's the structure. So let's see. Cell body. Processes are arising from that cell body. So if these processes are like this, like see, the branching part is there, right? The branching part is there, and see the cylindrical process, that will be your axon, these branches, and the axon is there. So that type of neurons are your multipolar neurons. The diagram that we make is of multipolar neurons. What is it? It is of multipolar neurons. Okay, bache. So we have the word dendron, right? So they are, what are they? Right, dendrons, dendrites. They are also the cell processes. These are branches. Yes, these are the cell processes. These are branches. So you can say they are short fiber which branch repeatedly and project out of the cell body. है ना? Yes, what are they? They are the short. Yes, what are they? Dendrons, dendrites. They are the short fibers, right? They are the short fibers that branch repeatedly. Right, what are these dendrons? They are the short fibers that branch repeatedly, and obviously they project out of the cell body, है ना? And they project out of the cell body. They are projecting out of the cell body, and they contain, bache, they contain what? Yes, they contain Nissls granule. Remember this thing. They contain Nissls granule. What do they contain? They contain Nissl granules. Is that clear? So now, bache, when you talk about a neuron, like see, that's how the things are. That's how the things are. Yes, you can see this neuron, you can see the cell body, soma, everything, dendrites. Now, see, we just said that there is a point-to-point connection in our body. What is there? Right? What is there? So they are having the point-to-point connection in our body, right? Point-to-point connection is there in our body. Just say one neuron, then next. So obviously it will transmit the nerve impulse. It will transmit that information, right, bache? It is like that. So when you look at this is one neuron, this is next. Let's say the terminal buttons here, the axon endings here, they'll pass the information to this neuron. So obviously dendrites are going to receive it, right? These dendrites are going to receive it. So dendrites, what are they going to do? Right, they carry the information towards the cell body. They carry information towards cell body. So you can take it like this, that they are at periphery, they are taking something towards the center. So obviously in their case, the conduction is centripetal. Centripetal from periphery towards the center. Right, now when you look at the axon, matlab that information will be, dendrites are going to receive it, they'll pass it to the cell body and then further to the axon. So we can say that axon is carrying the information away from cell body, right? It is carrying the information away from cell body. So what will be the word here? What type of conduction? This type of conduction will be there. Understood? This type of conduction will be there. Understood? Right, bache? Yes. Understood? So dendrons, they are going to receive the stimuli, they produce centripetal conduction, that is towards the cell body, but axon, they will carry it away from the cell body, so the centrifugal conduction is there. Okay, Okay, so when you talk about the axon, it is the longest neuron process having uniform diameter, right? It is cylindrical, and we know that it is having what? It is having uniform diameter. Yes or no? What does it contain? It contains uniform diameter, right? What does it contain? It contains uniform diameter. Understood? Understood? Yes. So longest cell process, diameter is uniform. Yes, बच्चे? Diameter is uniform, है ना? Yes or no? Yes or no? So now you know that from cell body it will arise. So there is a kind of this structure, na, some conical like structure, or you can simply say the structure from where the axon arises from the cell body. We call it as axon hillock. Axon hillock, and it is very, or you can say that most sensitive. It is the most sensitive part of neuron. Why? Because here you will see so many ion channels are there. So many ion channels are there. And bache, in the axon, neurofibrils and mitochondria present. So other organelles are absent here. Remember it. You will see neurofibrils, you will see mitochondria, but other organelles, they are absent. Right, bache? They are absent. Right, bache? They are absent. So this axon, the covering of axon is exolemma, and the cytoplasm here, for that you can use the word exoplasm. Right? You can call it as exoplasm. Understood? Yes? Understood? Yes or no? Yes or no? Yes or no? Right? So this is your axon, and then you know that the terminal endings are there. Yes, the terminal endings are there. Can you see that? Axon terminals, terminal endings are there. So this is your axon. And yes, bache, mainly this is the part who's going to conduct most of the function here in the neuron, right? So when you use the word nerve fibers, now you mainly consider the axon there, right? You mainly consider the axon there, okay? And this axon can be myelinated, means covered with myelin sheath, or it can be non-myelinated. It can be non-myelinated. We'll discuss that. So neurofibrils and mitochondria present. So even, I mentioned it already, but you can again write it, that Nizzel's granules are absent here. Right? They are absent in this exoplasm. Understood? They are absent in this exoplasm. Understood? Yes, they are absent in this exoplasm. Okay? Yes? Okay? Any doubt? Any doubt? So this is about the axon, the functional part of neuron. You can write it like this. What's that? It is the functional part of neuron. So now obviously we have different types of nerve fibers. We have myelinated, non-myelinated nerve fibers. We will discuss it, but up to this part all clear. So here you can see the terminal arborization. Here in the endings here you will see synaptic vesicles. What will you see? You will see synaptic vesicles, and these synaptic vesicles are fully loaded with what? They are fully loaded with neurotransmitters. They are fully loaded with what? They are fully loaded with neurotransmitters. Right, bache? Okay, they are fully loaded with neurotransmitters. Okay, done, right? So we'll discuss about it, but tell me, up to this part all clear? So now let's talk about the types of neurons, and here you guys can see the various types of neurons. So when you talk about the types, unipolar, uni mane single, right? So from the cell body, there is a single process only, right? The word is unipolar. Single process is arising from the cyton, right, bache? Single process is arising from the cyton. Only one process is arising, right? So example ke liye you have, for the example, what do you have? You have early embryos. You have early embryos of vertebrates and invertebrates. They contain such type of neurons. Okay, they contain such type of neurons, right bache? Right bache? Yes. So next is bipolar. So bi is the word, means two processes are arising from the cell body. Obviously two processes are arising from the cell body. So one dendrite, right bache? One dendrite, and one will be the, yes, one axon at one end, dendrite at another end. Axon at one end, dendrite at another. So it is a very important example, bipolar neurons, right? It's a very polar example. So you have retina of eye. Hope you remember. Now eye topic is deleted from your syllabus, but still, retina of eye, retina of eye, right? Retina of eye, right bache? Right bache? It is the retina of eye. Okay, so when you talk about the retina of eye, I don't know whether you know this or not. So retina, the image will form on it, no? So it is having photoreceptor cells. So that photoreceptor cells are having rods and cones, right bache? You have bipolar cells and one more layer is there. Please tell me about it. Right, just say photoreceptor cells, bipolar cells, and one more layer is there. Right, just say retina is made up of these three layers mainly. So please tell me what's that. Please tell me what's that. Retina is made up of you know the word? Anybody? Anybody? Not that difficult, yeah? Yes? Photoreceptor, bipolar, and ganglion cells. So basically you can say innermost is your. So light firstly will fall on this ganglion, then bipolar, then photoreceptor cells. Okay, right? So retina of eye, it is having the bipolar neurons. This example is important. This example is important one. Okay, other than that, olfactory epithelium, right? Other than that, olfactory epithelium. In olfactory epithelium also, you will see bipolar neurons are there. What will you see? You will see bipolar neurons are there. Okay. Okay. So retina, olfactory epithelium, they are having bipolar neurons. Okay, bache. Okay. Then the multipolar. Of course, this one, multipolar. So you will see it in. So see, from cell body, so many processes are arising, so many dendrites and axon. So here you will see it in cerebral cortex. Right, cerebral cortex. Now, bache, the word is pseudo unipolar. Pseudo means false. Pseudo means false. Right. So basically, it feels like that from the cell body only single process is arising, but then it will divide into two. Then it will divide into two. So what's that? What's that? It is a pseudo unipolar. Right. It is the pseudo unipolar neuron. So basically, where can you see this? You can see this in the dorsal root ganglia. Dorsal root ganglia of, yes, spinal cord. dorsal root ganglia of, yes bache, please tell me, please tell me. So it is the dorsal root ganglia of your spinal nerves. ठीक है, dorsal root ganglia of spinal nerves. So that's the example, you should know about it. Okay, these are the types of neurons. So you can see the diagram here, right? You can see the diagram here. We will also discuss the nerve fibers now. So I hope this part is clear, right bache? I hope this part is clear. Okay, sure. Yes. So on the basis of functions also bache, neurons are of different types, right? So जैसे when you are talking about this type, so it is on the basis of axon and dendrites, right? It is on the basis of axon and dendrites. On the basis of axon and dendrite, we are saying that neurons are of different types. Now bache, on the basis of function also, on the basis of function also, we have different types of neurons. For an example, your sensory neurons, also known as afferent one. You have motor neurons, also known as efferent one, and then interneurons, kind of mixing neurons, relay neurons they are. So on the basis of function also, you have three types of neurons. On the basis of what? On the basis of function also, you have three types of neuron. Right, bache? Yes. So when it is the sensory, the word is afferent. So here we have a trick, that is us, me. Us, me. That's the trick. Afferent, sensory, motor, efferent. So if you ever get confused, you can remember us, me, right? That is afferent, sensory, and motor is efferent. So as I said, it is the relay neuron as well as the connector neuron. Okay, chaliye. So sensory neurons, afferent, but they will carry that stimulus to the brain, yeah, to the central nervous system, to the brain and spinal cord. Okay, this is happening, what to do, what not to do, right? Sensory neurons. So they will carry that information to the, yes, they will carry that information, yeah, yes, they will carry the information to the central nervous system. So basically, in simple words, can I say that, matlab, who's going to sense that information? Our sense organs. So can I say that these neurons, they connect sense organs with central nervous system. CNS is your central nervous system, which includes brain and spinal cord. So obviously, they connect sense organ with yes, bache. They connect sense organ with the central nervous system. They will carry the information from sense organ to the central nervous system. Now motor neurons, E for efferent. So bache, this one, that's how you can define, connect central nervous system with effector. Matlab, it is like this ki, for an example, your sibling slapped you, right? So that information will pass to the brain. And then with the other hand, you are going to slap that person back, right? Then obviously, you'll be like this now. So basically, when your sensory neurons, afferent neurons, they will connect the sense organs with the central nervous system. They will carry that information from sense organ to the central nervous system, and that processed, right? They will carry that stimulus to the central nervous system, and that processed information, right, with the help of motor neurons, it will reach to the target organ. That's why we have used the word effector. And when you are talking about the interneuron relay or connector, obviously it is present in between. Yes, it is present in between sensory and motor neurons. It will present in between sensory and motor neurons, and definitely it helps in that transmission in between these two. Okay. Okay. So on the basis of function, you have three different types of neurons. Fine, bache? Fine. So now let's understand what is the meaning of nerve fibers. So firstly, tell me neuron clear. Neuron clear. So axon is the functional unit of neuron. That is why when you talk about the nerve fibers, mainly we are concerned with the axon part. Okay. So now you know what neuron is. Now you know what neuron is. Now, bache, your neuron, when the axon and dendrites, when this axon and dendrite, right, when these axons and dendrites of a nerve cell, when they are covered with, one or more sheets या two or more sheets माने covering, then we use the word nerve fiber for it. So basically your neurons are covering with the sheath. It is covering with a sheet. So nerve cell is covered with one or two, yeah, covered with so that is the nerve fibers. Then you will use the word nerve fiber. Is that clear? Then you will use the word nerve fiber. Is that clear? Yes. Nerve fiber word clear. So axon and dendrites of nerve cell, they are covered with one or more sheath. The word that we are going to use here is nerve fibers. Right, bache? The word that we are going to use here is nerve fiber. Right? Right? And nerves are what? Nerves. So they have different meanings. So what is the meaning of nerves? And yes, nerve fibers are of different type, myelinated or non-myelinated, medulated or non-medulated. We'll discuss that. So what are nerves? They are the group. So many nerve fiber together will form your nerves. Okay, bache? So they are bundles of nerve fiber. They are bundles of nerve fibers, means so many nerve fibers together. So they are bundles of nerve fiber in peripheral nervous system. Right? They are the bundle of nerve fibers in peripheral nervous system. Yes, bache. They are the bundle of nerve fibers in peripheral nervous system. Doubt? Yes? Any doubt? They are the bundle of nerve fibers in peripheral nervous system. Any doubt? Yes, bache. And see, one more thing is there. There is one word track. Track. In the brain you will read it. So when the bundle of nerve fiber, it is the collection या bundle of nerve fibers, मतलब that bundle of nerve fiber in central nervous system. In central nervous system you will call it as track. Right? But in the peripheral nervous system, right, in the peripheral nervous system, you will call it as nerve. In the central nervous system, you will call it as track. Is that clear? Is that clear? Yes? Yes? Done? Okay. Okay, so now we also use the word ganglia, right? We also use the word ganglia, right? So ganglia is the word and there is one more word, that is nuclei. Okay, so ganglia, it is composed of nerve cell, it is composed of your cell body, means aggregate of cell bodies. So many cell bodies together, they form the ganglia, and we use this word in PNS. We use this word in PNS. Is that clear? Is that clear? We use this word in PNS, ganglia. Okay, so it is the collection of, right? It is the collection of cell bodies. Yeah, it is the collection of cell bodies. Yeah, composed of cell bodies. Okay, ganglia is composed of, matlab it is having so many cell bodies. And nuclei term, it is the collection of cell bodies, or you can say that cytons in CNS. So is it clear? Just say when we talk about the brain, we say na two cerebral hemispheres are there and they are connected with a track of nerve fibers known as corpus callosum. Right? There is a track of nerve fibers that is known as corpus callosum, that is known as corpus callosum. Track of nerve fiber is the word in the NCERT. So track mane, it is the bundle of nerve fibers only. So bundle of nerve fibers in the case of central nervous system is known as track, and the bundle of nerve fibers in the case of peripheral nervous system is known as nerves, right? It is known as nerves. Okay bache, so track, nuclear, all these words clear? Yes. Is it clear? The types clear? So now let's discuss about the myelin, myelinogenesis, that is myelin formation. Right, bache? Right, bache? So myelin. So what is this myelin? What is this myelin? It's a fatty layer. It's a fatty layer, right? Fatty layer. It is acting like an insulatory layer. Just let's say ki wire is covered with a plastic, na? Right? That acts like an insulatory layer. Same is the case here. So when you talk about the myelin, right, it's a fatty layer with a high electrical resistance, right? With a high electric resistance and acts like an insulatory layer. And acts like an insulatory layer. Okay. Okay. Done, bache? Yes. Yes. So you can say myelin is having lipid, protein and water. It is what? It is having lipids, proteins and water. Lipids, proteins and water is making the myelin. It is making the myelin. Okay. Okay. So we have, yes, we have myelinated nerve fiber or we have non-myelinated nerve fibers. Yes, bache. We have myelinated nerve fibers and we have non-myelinated nerve fibers. So we have to discuss about it. Okay. Okay. Done? Chaliye. So medulated is also the word that we use. That's what we have. So obviously medullary sheath is present, right? Medullary sheath is present. Wait, it's like this. It's like this. Okay, just imagine just imagine this part na, yes, this part of, imagine it as an axon. So this part here is covered with, right? Here you have the myelin sheath. Here you have the one more covering myelin sheath. Now let's say there is a gap in between. Let's say, okay, let's say there is a gap in between. There is a gap in between. Then again it is covered here. Okay, again it is covered here. So that gap is, you know that already, right? That gap, you know that already. It is NOR, nodes of Renware. And this is your myelin sheath, right? This is your myelin sheath. Is that clear? And this one is non-myelinated, right? This one is non-myelinated. non-medulated. Okay, this one is non-myelinated, this one is non-medulated. That is how it is. So when you talk about the myelinated nerve fibers, they are generally in the white matter of brain and spinal cord, and in the spinal and peripheral nerves also. Right? Where are you going to see it? In the white matter of brain and spinal cord, and in the cranial nerves and spinal nerves. So if cranial nerves and spinal nerves are involved now, students, if cranial nerves and spinal nerves are involved, so we can say that it is there in the peripheral nervous system. Right, bache? Peripheral nervous system. Okay, and non-myelinated one, medulated one, non-myelinated one, they are in commonly in ANS, autonomic nervous system, and somatic neural system, nervous system. So they are commonly found in autonomic nervous system and somatic neural system. Okay, autonomic nervous system and somatic neural system. Is that clear? Is that clear? Is that clear? Sure? Sure? Sure, बच्चे? So they are in white matter because these myelinated nerve sheaths, they appear white in fresh state. And this one appears gray. So that's the point. And now see, because neurons, they are associated with what? These neurons, they are associated with the conduction of nerve impulse, right? Just let's say it is a myelinated nerve fiber. Okay, so when it comes to the transmission of information, so obviously it is acting like, this layer is acting like a layer of insulation. How is it acting? It is a layer of insulation, insulatory layer. So ions will not be able to move here and there, right? Because it acts like an insulatory layer, right? It acts like insulatory layer. Right, it acts like insulatory layer. So now, in this case, when there is the transmission, let's say this is the point. Let's say this is the point from here to here, here to here, here to here. So it is saltatory conduction. Saltatory conduction, right? So nerve impulse will jump from this point to this point to this point to this point. So it's fast, right? It is fast. It is fast. But in non-myelinated nerve fibers, it will be slow. But in non-myelinated nerve fibers, it will be slow. Right, bache? Right, bache? In the non-myelinated nerve fibers, it will be slow. Okay. And here you guys can see, this is your myelin sheath. In this diagram, you people can see. In this diagram, you people can see. This is the node of Renvier. Here you have the myelin sheath. Look at the covering here. 1, 2, 3, 4. Look at this covering. Right, and yes, there is word Schwann cell. So now this is what we need to discuss. Right, now this is what we need to discuss. So you can see here, you have myelinated sector, Schwann cell is there, myelinated one in between Renvier interval or node of Renvier. Right, so let's talk about these Schwann cells. Let's talk about the oligodendrocytes. Okay, let's talk about these Schwann cells. Let's talk about these oligodendrocytes. So any doubt? Yes. Pranitha, any doubt? Any doubt? Yes, please. Done? Chaliye. So, bache, it is said that each segment of myelin sheath, obviously it's a type of cell, we will discuss that also. So each segment of myelin sheath is made up of Schwann cells, Oh, SCH. WA. Schwann cells. Write down. Each segment of myelin sheath is formed by Schwann cells. Please write down. Yes, bache. Please write down. So they take part in the formation of this myelin, right? Which will not allow the ions to get leak, right? Which will act like the layer of insulation. Okay. Okay, okay, okay. Right. So it is the Schwann cells that are going to make it. So each segment of myelin sheath is made up of, formed by, my bad. It is formed by Schwann cells, and that too in PNS. Remember this thing. That too in PNS, in peripheral nervous system. But bache, in CNS, in central nervous system, I told you in central nervous system, white matter there, right? It is having the modulated nerve fiber. So in CNS, oligodendrocytes. In CNS, there are another cells, oligodendrocytes. They form myelin sheath. It's a very important point, right? They are the one that form myelin sheath, right? So in CNS, oligodendrocytes, they form myelin sheath, right? In CNS, oligodendrocytes, they form myelin sheath. Understood? They form myelin sheath. Okay? Done, bache? So oligodendrocytes are going to form it. It is a very important question. So I can say, so oligodendrocytes are nothing. They are the type of neuroglial cells. So in the central nervous system, we don't have the Schwann cells. So oligodendrocytes will do myelinogenesis. Right, bache? And yes. One more thing. Just let's say this is the myelinated nerve fiber, right? So we are saying Schwann cells are forming it, right? Schwann cells are forming it. So remember one line here, bache. Remember one line here. Ki myelin sheath, okay? Myelin sheath, yes. That this myelin sheath is covered by this medulla, or myelin is covered by a membrane, by a thin membrane, that is also known as sheath of Schwann cells. So basically your Schwann cells are covering this medulla here. So it is covered by thin membrane, that is also known as sheath of Schwann cells, right? But and there is another word for it, that is known as sheath of Schwann cells. The medulla is covered by this thin membrane, thin cell membrane, sheath of Schwann cells, and neurilemma. These are the two words that we use. Neurilemma. These are the two words that we use. Okay? These are the two words that we use here. It is neurilemma or sheath of Schwann cells, right? So medulla or myelin is covered by a thin membrane. It is covered by a thin membrane, that is the sheath of Schwann cells, right, bache? That is the sheath of Schwann cells, and it is also known as neurilemma. It is also known as neurilemma. Understood? Pakka? Done, bache? Okay, so myelin sheath is covered by thin membrane known as neurolemma or the sheath of Schwann cells, known as neurolemma or sheath of Schwann cells. Is that clear? Is that clear? Yes, bache, is that clear? Now you tell me, if there is a non-myelinated nerve fiber, what do you think? Non-myelinated nerve fibers, do they contain the sheath of Schwann cells? Non-myelinated nerve fibers, what do you think? Do they contain the sheath of Schwann cells? Do they contain sheath of Schwann cells? Tell me. Yes? So Schwann cells, they are helping in the deposition of myelin sheath. Now you tell me, if there is a unmyelinated nerve fiber, do you think the sheath of Schwann cells will be there? I told you that non-myelinated nerve fibers, they are commonly found in ANS and SNS, मतलब somatic neural system and autonomic neural system, right? Somatic neural system or autonomic neural system. So here what you people are going to see, मतलब it is also the part of your PNS only. So what you people will see here, that they are, right, they are non-myelinated. So in their case, what do we have? Do we have the layer of Schwann cells? Yes, there will be. There will be a layer of Schwann cells, but that Schwann cells are not forming the myelin sheath. Let me mark it here in your NCERT. Okay, let me mark it in NCERT. Ye dekho. See this. Okay, so synaptic knob, neurotransmitters, we have discussed it. Okay, so multipolar, bipolar, unipolar, done. So when you talk about the myelinated and non-myelinated nerve fibers, so myelinated nerve fibers are enveloped with Schwann cells. Myelinated nerve fibers are enveloped with Schwann cells, which form a myelin sheath around the axon. So the gaps between two adjacent myelin sheath, it is the nodes of Ranvier, right? And myelinated nerve fibers are very common in your spinal and cranial nerves, right? They are very common in your spinal and cranial nerves, right? Even in the white part of your brain and spinal cord. But here, look at this line. This is important. So even the unmyelinated nerve fiber is enclosed by Schwann cell that doesn't form a myelin sheath, right? They are around the axon and is commonly found in autonomous and somatic neural system. So this part is important, right, bache? This part is important. Okay, do it. Yes, this part is important. Okay, neurolemma clear? So outside the myelin sheath, there is a layer of Schwann cells, that is neurolemma, right? The sheath of Schwann cells, and that sheath of Schwann cell is also continuous over nodes of Ranvier. So for neurolemma, you can write down it is even continuous over the nodes of Ranvier. Okay, it is even continuous over the nodes of Ranvier. Is that clear? Is that clear? It is continuous over the nodes of Ranvier. Done? Yes? Okay. Any doubt? So now let's discuss about the other type of cells, right? There is one more diagram that I have added. Just let me check. Here, look at this. So basically it is showing that What is it showing? It is showing our blood-brain barrier, BBB, right? Blood-brain barrier. So basically you can see the microglial cells, astrocytes, right? Astrocytes, microglial cells. Look at the neurons there. Look at these pericytes, right? The leukocytes there, the endothelial cells there. That type of system is there in our blood. Matlab it is showing the blood-brain barrier. Right now we have to discuss it, but before that we will talk about the other type of cells which are present, right? What are we going to discuss, bache? We will talk about the other type of cells. You cannot say that in the nervous system you have neurons only. No, neurons are there, but there are some other type of cells also. Let's discuss that, and that are your neuroglial cells. That are your neuroglial cells, right? Even in the chapter structural organization in animals, right? Even in structural organization in animals, it is given na, about the neuroglial cells. They are the supporting cells. Yes, what are the? They are the specialized cells found in the brain and the spinal cord, and they support the neuron and their fiber. They are the specialized cells found in nervous system, and they support, they are the specialized cells that are found in nervous system, and they support neurons and its fibers. Okay, so we have different types here. What do we have here? We have different types of neuroglial cells. So you remember, they comprise 50% of all the nervous brain cells. Comprise 50% of all the nerve cells. Right, bache? So you have astrocytes there, right? Remember, astrocytes there. You have oligodendrocytes, right? You have oligodendrocytes there, and you have microglial cells, right? Microglial cells, that's the word. So remember, microglial cells, जैसे liver is having Kupffer cells, the phagocytotic cells of liver, they are the Kupffer cells. So same way, when you are talking about the microglial cells, they are the phagocytotic, cells of your nervous system. So bache, if they are the phagocytotic cells of nervous system, obviously if there is any foreign particle there, they are going to kill it, they are going to destroy it, right? So they are the phagocytotic cells of the nervous system, your microglial cells. Understood? Yes, bache. So you know that oligodendrocytes. So basically, as I said, that these cells are going to support the neurons. They help in their repair as well. Just say astrocytes, they play the role in the repair of the nervous system, of the neurons, right, bache? So microglial cells, they are phagocytotic cells of the nervous system. They are also obviously phagocytotic, is it? It is. So we can also use the word squangers. And oligodendrocytes, they are smaller cells having few processes. And how do they help? They help in myelin sheath formation, and that you know in central nervous system, right? So oligocytes, how do they help? They form myelin sheath in central nervous system. So be specific. What are you talking about? You're talking about the central nervous system. And then you have the astrocytes. What do you have? Astrocytes. They form, bache, I told you about the blood-brain barrier. They are going to form the blood-brain barrier, right? They even repair. They help in the repair of your nervous system, right? Right, bache, of your neurons. Okay. So what is the word? Yes, bache. Yes, what is the word? The word is astrocytes. Okay. So say provide mechanical support, right? They form blood-brain barrier, right? They play a role in maintenance of blood-brain barrier and in the repair of the damaged area of nervous system. ठीक है, help in the repair of damaged area of nervous system. Understood? Yes. So what is the meaning of blood brain barrier here? Right बच्चे, right बच्चे, it's simple, right? You have the blood vessels, you have the blood vessels here, you people can see, right? So with that blood vessels your astrocytes are attached, and that too with the help of tight junctions. ये देखो, you have the astrocytes, you have the blood cells there, so ये देखो, they are associated with it with the help of these tight junctions. So they will not allow the neurotoxins to enter there, right? They will not allow the new entry of neurotoxins basically, right? So these astrocytes, they are coupled with what? They are coupled with the tight junctions, right? They are coupled with the tight junctions. Of course, even this is the capillary epithelium. What is this? This is the blood capillary epithelium. So it will not allow the neurotoxins to enter. It will not allow the neurotoxin to enter. So look at the setup here. These are astrocytes, microglial cells are also there. This is the blood capillary. So they will not allow any neurotoxin to enter. They will not allow any neurotoxin to enter. So that is how it works. So this is all about the different types of cells there, right? This is all about the different types of cells there. Is that clear? Is that clear? So neurons, clear, right? What is efferent? Like now you already know what are efferent neurons. So obviously efferent fibers, they are the sensory fibers. You know their role. I don't think we need to discuss. So let's make this flow chart and then let's discuss the things further. Okay, so when you talk about the neural system or the nervous system, what do we have? We have central neural system and we have peripheral neural system. Am I right? We have central neural system, we have peripheral neural system. Central neural system is there, peripheral neural system is there. So central neural system matlab brain and spinal cord, right? Brain and spinal cord. Is that clear? Right? What is it? It is the brain and the spinal cord. And what do you have there in bache? Yes, what do you have there in this peripheral nervous system? It is having your spinal nerves, it is having your cranial nerves. So if you check the syllabus, they are the part, right? If you check the syllabus, they are still the part. So tell me, what will you see there? What will you see there? Obviously, autonomic neural system. And this autonomic neural system is what? It is involuntary. What is the meaning of involuntary? We cannot control it with our own will. And then you also have somatic neural system. Yes, bache, what do you have? You even have somatic neural system. And that somatic neural system is what? It is voluntary. We can control it with our own will. So when you talk about this bache, autonomic neural system, you talk about the sympathetic and parasympathetic. Sympathetic nerve fibers are there and parasympathetic. Nerve fibers are there. Are you getting my point? Sympathetic nerve fibers, parasympathetic nerve fibers are there. So sympathetic deals with fear or flight wale emotions. It deals with fear or flight kind of emotions. And parasympathetic, rest and digest. Rest and digest. And that's what we will discuss in detail. That is what we have to discuss in detail. Any doubt? Any doubt? So neural system, central one will have brain. It will have spinal cord. So from this brain, you know that the cranial nerves will arise. From spinal cord, spinal nerves will arise. Peripheral nervous system, autonomic, somatic. Autonomic is involuntary. We cannot control it with our own will. So here you have sympathetic nerve fibers, parasympathetic nerve fiber, somatic neural system. Voluntary it is. So here you are talking about your skeletal system, because we can control it with our own will. We can control it with our own will. Is that clear? Is that clear? Yes. So when you talk about the viscera, mane body organs, you can take the example of your Digestive system. Your digestive system, the hollow wall of your alimentary canal is having the nerve fibers. It is also having the nerve fibers, and that is also autonomic. So when you talk about the visceral nervous system, it is the part of PNS only, bache, right? And it is having the complex of nerves, even the fibers, ganglia and plexus. So whenever you read the word plexus, mane network. It is a network of nerve fibers. Right? When the word is plexus, what is it? It is the network of nerve fibers. It is the network of nerve fibers. Is that clear? Is that clear? Yes? Is that clear? So visceral nervous system is the part. Yes, it is the part of peripheral nervous system that comprises the whole complex of nerves, fibers, ganglia, and even the plexus by which impulses travel from CNS to the viscera and from viscera to CNS. Any doubt? Any doubt? So, neurons, done. Diagram, done. Nerve impulse conduction, we have to understand. But before that, let's discuss Upta Fagwasha. Do you know that, bache? What is this Upta Fagwasha? Do you know that what is this Upta Fagwasha? So when you talk about your syllabus, CNS is mentioned in your syllabus. So central nervous system is there, brain is there, spinal cord is there. Matlab, when you read your syllabus from the official information bulletin, I hope you guys have seen that. I hope you guys have seen the official information bulletin. So when you people see this, yes, when you people see this, so the brain, the central nervous system, the peripheral nervous system is given there. The central nervous system, the peripheral nervous system is given there. So obviously when PNS is there, right? When PNS is there means peripheral nervous system, cranial nerves, spinal nerves, they all will come. Yes, they all will come. Yes or no? They all will come. So obviously we have to discuss it. Right? Right? Okay, है ना? So that is also somewhere the part of your syllabus. Okay, right, bache, right? So let's discuss that. Let's talk about the cranial nerves. I'm not saying that remember it in detail, but if you can, you should. You should. We have the tricks for that, and that is very simple. And the trick here is, it's a mantra that you need to chant. Upta ya utata fagva sha. Right? You use the first letter here and then upta fagva sha. Upta fagva sha ya utata fagva sha. Upta fagva sha, or you can call it utata fagva sha. Upta fagva sha, or you can call it utata fagva sha. So then is all up ak. That's my way to remember. Right? So cranial nerves you are talking about, they are 12 pairs. How many cranial nerves are there? 12 pairs. 12 pairs means total 24. So it is upta fagva sha ya utata fagva sha. Upta fagva sha ya utata fagva sha. So olfactory, optic, oculomotor, pathetic, right? Trigeminal, abducens, then facial, then auditory, then glossopharyngeal, then vagus, spinal accessory, and hypoglossal. And hypoglossal. Understood? Understood? So what is the word here? It is upta fagva sha. All up ak. All up ak. Okay? Upta ya utata. Why utata? I'm writing it again. It's all up ak. Olfactory, optic, oculomotor. So pathetic is also known as trochlear, right? Then trigeminal is there. Then abducens, of course, facial, auditory, glossopharyngeal, vagus, spinal accessory, hypoglossal, right? 12 pairs. Utterfagvasha, right? Utterfagvasha, or you can call it as Utterfagvasha. That's how I remember it. Okay, that's how I remember it. Okay, yes, yes, done. So जैसे vagus is also known as pneumogastric. So I'm not saying that yes, remember it, right? If you can, you should. If you are targeting 360 out of 360, go for it. But yes, from here you should know which one is sensory, right? And which one is your motor, and which one is playing the function of both. So this is what we have. This is what we have. ठीक है? Utterfagvasha, right? Upta or Utterfagvasha. ठीक है? So see, look at this. Look at this and their function. जैसे in the paper they can ask you what is the role of parasympathetic or sympathetic nerve fiber. That is also associated with this PNS. Now we have to discuss it. So all I want to say is yes, this is what you can remember. 12 pairs are there. The formula is Utterfagvasha or Utterfagvasha. Wait, diagrams. ये देखो जैसे brain is there, spinal cord is there, right बच्चे? Brain is there, spinal cord is there. Okay बच्चे, जैसे ये देखो, this is your brain box now, that complete skull, vertebral column, they are protecting your spinal cord. From spinal cord, spinal nerves are arising. From your brain part, you have the, right? From your brain part, you have the cranial nerves. Sorry, you have the cranial nerves. So yes, go for the trick. So there are many, जैसे here, this image I got from the Google, of course. So जैसे only one of the two athletes felt very good, victorious and healthy. Only one of the two athletes, they felt very good, victorious and healthy. So that is also the thing, but I remember it like this. Utterfagvasha. Utterfagvasha. Jैसे another trick here. Some say marry money, but my brother says big brains matter most. Some say marry money, but my brother says big brains matter the most. Matters most. You have to be specific. Some say marry money, but my brother, once again, I'll write it here. Chalo, let me write it properly. So the trick is Upta ya Uttata Fagvasha. Right? Uttata ya Upta Fagvasha. So all up ak, olfactory, optic, obviously related to vision na, oculomotor. ठीक है? P or T. मतलब it can be, you can call it pathetic or trochlear. One and the same thing. Then trigeminal. Auditory is having two types, but let's not focus there. Auditory, right? Then facial is there. Sorry. My bad. Trigeminal. Then you have abducens, facial, auditory, glossopharyngeal, vagus, also known as pneumogastric, spinal accessory, or hypoglossal. So it is 1, it is 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 or 12. So some say marry money, but my brother says big brains matters most, right? So what is the meaning here? S sensory, S sensory, M motor, again M motor, B माने both, मतलब they have both the roles, sensory plus motor. Okay, my motor. Brother means both. Say sensory. Both, motor and motor. So that's the trick to remember. So one is upta bhagwasha, the another one is this. Right, bache? The another one is this. Okay? So both mane mixed, sensory and. So can I say that 1, 2 and 1, 2 and your which one? Eighth one? Pair 1, 2 and it's a pair of sensory nerve fibers. The sensory nerves. Yes, sensory nerves. 3 pairs are sensory. Then motor. 1, 2, 3, 4, 5. Right? 5 pairs. Yes, bache. 5 pairs are motor nerves. 3rd, 4th, 6th, 9th और 12th ना. ये देखो. Sorry, 11th और 12th ना. 1, 2, 11, 12th. Now there will be no confusion. Okay. So motor के लिए 3rd, 4th, 6th, 11th, 12th. 1, 2, 3, 4, 5. 5 pairs motor. And obviously rest of it mixed. 1, 2, 3 or 4. They are mixed. Okay, they are mixed. Okay, yes, they are mixed. So this information is important. Right, so Upta Phagwasha, some say marry money, but my brother says big brains matters most. Some say marry money, but my brother says big brains matter most. Okay, okay, right. So yes, I'm not saying that very frequently. Right, that very frequently they ask the question from this part, of course not, but yes, you should know about it. Okay, you are going to be a doctor na, so you should have that aptitude. So ye dekho, their functions also, matlab not important, but olfactory related to smell, optic related to vision, oculomotor serves muscles of the eye, trochanter serves the superior oblique eye muscles, trigeminal, sensory from face and mouth, motor to muscles of mastication because it is having the mixed function, no? Mixed function na, both motor and sensory. So I'll share the PDF in the Telegram group. You will get the idea. So look at this part. Look at this part. That's the brain. These are the 2 hemispheres. Right. Right. And there will be a track of nerve fiber. That is corpus callosum, which is going to connect it. So you can see the nerves here. Olfactory, oculomotor and all. Right. Ye dekho. The nerves, the way they are arising. Look at this. ठीक है, डन बच्चे, डन. ये देखो, this part of brain and the way the nerves are arising. Okay, brain and the nerves are arising. So obviously there will be go to the different parts and they are going to control the functions. So this slide and this slide is also having the function. Olfactory nerve, location is nasal epithelium, sense of smell, optic nerve, retina, vision, oculomotor. It is in the midbrain. Right, बच्चे, eye movements के लिए it is helpful. So that's it. ठीक है? That's it. So let's have a break. Let's meet at 9 PM. What say? ठीक है? Let's have a break. Break time. Okay? Hi guys, welcome back. Hello, welcome back. So yes, done with this Upta Pagwasha thing? Hi guys. So are you done with this Upta Pagwasha thing? Right? Okay. So this is about the cranial nerves. Okay. So same way spinal nerves are also there. Right? Spinal nerves are also there. And yes, am I audible? Yes, please. Am I audible? Please tell me, am I audible? Okay. Okay, so when you talk about the spinal nerves, we have total 31 pairs, right? So I hope you remember CTLSC, that is cervical, thoracic, lumbar, sacral, coccyx, right? So like we have these now, vertebrae, CTLSC, CTLSC. So same way here we have, right, same way here we have the spinal nerves, 8 pairs, right, 7, 12, 12 pairs. Yes, bache, 8 pairs, 12 pairs, 5 pairs, 5 pairs, and 1 pair. Right, so you have the 31 pairs of spinal nerves as well, right? 31 pairs of spinal nerves as well. Okay, so now let's discuss the, so ye dekho, yes, okay, spinal nerves and their function, 8 pairs, 12 pair, 5 pair, 5 pair, 1 pair. Jaise C1 to C4 will help in breathing, in head and neck movement, heart rate, shoulder movement. So that's how they work. Okay, chaliye. So now bache, we will talk about this. You know the structure of neuron. Now we will discuss about the generation and conduction of nerve impulse. What are we going to discuss now? Generation and conduction of nerve impulse. But before that, let's understand this part, right? Let's understand this part. Let's get to know about the neurons. Obviously, okay. So firstly, generation and conduction of nerve impulse, right? And that we are going to study under the topic of ionic theory. But before that, right? Before that, you need to know about the neurons. That what kind of cells are they? Right? You need to know about what? You need to know about the neurons, that what kind of cells are they? Obviously, they are the excitable cells, right? What kind of cells are they, guys? What kind of cells are they? They are the excitable cells, right? They are the excitable cells. And yes, how can I forget? When I was teaching you this part now, cells, right? Yes, I missed that thing, neurosecretory cells. Yes. Here I missed the neurosecretory cells and ependymal cells. Neurosecretory cells will form neurohormones, right? Neurosecretory cells are also there. They'll form neurohormones, if you remember, right? Bache, they are there in the hypothalamus, right? Bache, they are the endocrine cells. Other than ependymal cells are there. I'm writing the word here. Ependymal cells are there. So, bache, in the brain, like we have the cavities. For the cavities, we use the word ventricles. Ventricles. So that ventricles are lined with these ependymal cells, and these ependymal cells, right, they are basically playing role in what? These ependymal cells will also play role in, yes, my dear students, right, they are lining the ventricles. They have many villi and cilia, so they help in absorption and flow of cerebrospinal fluid. Okay, they help in absorption because cilia is also there. So they help in absorption and flow of, right, bache, cerebrospinal fluid. That's it. Okay, that's it. So now let's discuss about the generation and conduction of nerve impulse. So when you talk about the neurons, yes, generation and conduction of nerve impulse. What's that? Generation and conduction of nerve impulse. So first thing that we need to know is that what is the meaning of excitable cells? Anybody in the class? What is the meaning of excitable cells? Anybody in the class? Yes? Yes, please. Yes, please. What is the meaning of excitable cells? Excitable cells. Anybody in the class? Obviously neurons, they are the excitable cells because their membranes, right? You're talking about the neurons here. They are the excitable cells because their membranes are in polarized state. Right, bache? Why? Because their membranes are in polarized state. Their membranes are in polarized state. Why? Let's say you know that axon is the functional, right? Functional part of neuron. So here the ion composition and in the surrounding, right? Here the ion composition and in the surrounding, the ion composition, it varies, right? It varies. And these membranes, they are in this polarized state. Okay, so neurons are excitable cells. So before knowing this, before understanding this, you need to know that when you talk about the neural tissue, when you talk about the nervous tissue or the neural tissue, it is having the property of excitability and conductivity. When we talk about the nervous tissue, what do we have in the nervous tissue, guys? Yes, when you talk about the nervous tissue, right? So here you will see two things are there. The property of excitability, and the property of conductivity. These two things are there in the nervous tissue. The property of excitability and the property of conductivity. So when I'm saying excitability means nerve cells can generate impulse in response to a stimulus. Right, in response to a stimulus, that is making them special. Right, when we talk about the transmission of nerve impulse, when we are talking about the nerve impulse, we are talking about the neurons. There is a reason behind, now. There is a reason behind if we are talking about the neurons. There is a reason behind if we are talking about the neurons. Of course, there is a reason, right? And what is that reason, guys? Obviously, neurons are special cells. They have the special property. Their membranes can do that. What can they do, right? When you talk about the neurons, you will see the nervous tissue. It is the neurons. It is the ability of, yes, it is the ability of nerve cell. Yes, my dear students. It is the ability of nerve cells to generate an yes, bache, to generate an electric impulse in response to, yes, in response to stimulus. Right, whenever the neurons, the nerve cells, whenever they get the stimulus, they can generate the electric impulse. How? By altering the potential difference across their plasma membrane. How? By altering the, like, let's say, yes, let's say when a neuron is not conducting, the condition will be like this. I'll explain it. And when it will be conducting, the condition is going to be like this. Why? Why? Right, because neuron cells are having this property. They show this electrical excitability, right? They show that, right, they can generate the electrical impulse in response to stimulus by altering, yes, bache, by altering the, what can they alter? Potential difference. By altering the normal potential difference across their membrane. By altering the normal potential difference across their membrane. Across their membrane. That is the excitability. Now when the conductivity is the word, right? Right? Yes, so obviously they'll get excited. They'll get excited, right? They can generate that electrical impulse. How? How? By the movement of ions. Right, bache? By altering the normal potential difference across the membrane. And it is not the only thing, they can even conduct it. Means they have generated the electrical impulse, and now they can even what? They can even transmit that electrical impulse. Are you getting my point? Right? So the ability of nerve cell to rapidly transmit the electric impulse, right? So it is ability of nerve cell to transmit the electrical impulse. Obviously from the site of its origin throughout its length, है ना? From the site of its origin to, right बच्चे, along their length, right? Chalo, let me write it. In a particular direction across the length. Okay, so this is the special property of the nervous tissue. This is the special property of the nerve cells. Is that clear? This is the special property of the nervous tissue. This is the special property of the nerve cells. Now we have two words. One is resting potential. Yes, students. One is resting potential. Another is action potential. The name is making it very clear. When your neurons, when they are not conducting anything, when your neurons, right, when the neurons, they are not conducting anything. Yes, bache, when the neurons, they are not conducting anything, they are not conducting any nerve impulse, right, then that membrane potential will be the resting potential. When it will conduct the nerve impulse, that membrane potential will change because the ionic movement will be there, because the sodium influx will be there, then it will come as the action potential, and obviously the value will also change, right? The value will also change. So resting potential means your membrane is polarized. Resting potential means Surbhi, the membrane is polarized, and I'll tell you why. So resting potential mein resting nerve fiber which is not conducting, it shows a potential difference between inside and outside, obviously. Right, bache? Obviously. Right, bache? Right, bache? So I am not going to write the definition, proper definition here. You please understand it directly. So resting potential means your neuron is not conducting. Your neuron is not conducting. And I told you, they are the excitable cells. Right, they are the excitable cells, right? Different ion concentration is there. So when you are talking about the resting potential now, when you are talking about the resting potential now, outside will be positive, remember, because like extracellular fluid is having more positive ions, it will be positive, inside will be negative, and you use the word polarized. Means when your neuron is not conducting any nerve impulse, right, then what will be the scenario? What will be the scenario? This will be the condition, this will be the scenario, outside positive, inside negative. So extracellular fluid is having more positive charge, extracellular fluid is having more positive charge, extracellular fluid is having more positive charge. So outside will be the plus, inside will be the minus, and I will tell you why. I will tell you why. So it is somewhere in between minus 50 volts to 100 millivolts, right? Right, if you talk about its value, so you can say that it can be in between minus 50 to 100 millivolts, minus 50 to 100 millivolts. So membrane is electrically polarized, right? So somewhere it will be in between this, and generally we say minus 70 MV, millivolt, you know about it. Now the second thing is action potential. Right, your neuron is in action. It is transmitting that impulse. It is transmitting that impulse. Why? Because it will receive the stimulus. We'll talk about that stimulus, all or none principle, summation principle. So many things are there. But let's say at this point, when it will receive the stimulus, obviously its value should be above threshold stimulus. So ions will come in. Inside will become positive. Outside will become negative. So it is depolarized state. It is the action potential. It is a depolarized state. So if you remember when I was teaching you heart, I was telling you about the contraction, relaxation. I told you when it is the contraction, it is systole, it is action potential, it is depolarized. Right, depolarized. Ventricles depolarization means ventricular contraction is there, means ventricular systole is there. Right, bache, that's how you can remember. And do you know how do I remember? My funda is very simple here, that when you are at rest, you're not doing anything, you're wasting your time, you feel negative, है ना? We feel negative, we don't feel good, we feel like, yeah, why is it so, why it is happening to me? That's how I remember. So when there is a resting potential, inside will be negative, right? When you are doing something, when you have decided, okay, these are my targets, I need to fulfill my targets, obviously, and when you'll do it, you'll feel positive. That's why I remember. And this is the depolarized state. This is the depolarized state. This is about the resting potential and the action potential. Okay? Right? 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 So how do we define the nerve impulse? How do we define the nerve impulse? It's a wave. Right? How do we define a nerve impulse? What is it? It's a wave of reversed polarity or depolarization. It's a wave of reversed polarity or depolarization, obviously across the length of axon. Yes or no? Yes or no? So it is a wave of reverse polarity or depolarization moving down an axon, right? Moving down an axon. So what's that? What's that? It is your nerve impulse. What's that? It is your nerve impulse. Understood? So when the neuron will be stimulated, electrical disturbance will be there, right, bache? And that's how the things are going to be, and we'll explain it. How are we going to explain it? We are going to explain it with the help of Ionic theory, right? We are going to explain it with the help of Ionic theory. So it is one of the most important topics. Is that clear? It is one of the most important topics. Is that clear? Is that clear? Yes? Okay, so what is it? It is the wave of reversed polarity, right? So now if I have to tell you the properties of action potential, if I have to tell you the properties of action potential, yeah, features of action potential. So number one is threshold stimulus. So I know you understand this. What is it? When you are using the word threshold stimulus, what is the meaning of that? What is the meaning of that? Yes? Yes, threshold stimulus. I am saying that your neurons, this is the membrane here. It will receive a stimulus. It's not like that, but any stimulus can generate the action potential. Any stimulus can cause the disturbance. It is not like that. Any stimulus can cause the disturbance. It can cause the action potential. No, not at all. Right? Not at all. Any potential cannot. It's not like that. Any potential can. Right? Any potential can generate the nerve impulse. No, there is a minimum intensity of the stimulus that is required, right? There is the minimum intensity of stimulus that is required, right? That minimum has to cross. Okay, bache? So it is of certain minimum intensity in order to produce an action potential. So what is this threshold stimulus? That a stimulus must be of certain minimum intensity. It should be of certain minimum intensity to produce an action potential. So that minimum value is your threshold, right, bache? Like to qualify an exam out of 100, you need to score minimum 35 marks. If you are not scoring 35 marks, you will not be considered as pass, right? Okay. So that 33 or 35 marks are what? That 33 or 35 marks are what? That 33 or 35 marks are what? That is basically that threshold that we need to cross, we need to achieve. Is that clear? Is that clear? Yes? Right? Okay. Okay. Okay, chalo. So second point, I'm writing the properties here, then I'll explain. It is of summation and then all or none principle. All or none principle. Let me explain that. So threshold stimulus, right? So if below threshold stimulus, the action potential will not be generated. It has to be above threshold stimulus, right? Now let's say there is a stimulus, right? This stimulus is a sub-threshold. It is below threshold. It cannot generate the action potential, of course. So let's say if there will be many that type of, you know, stimulus, sub-threshold stimulus. So their summation means sum, means total, means add-on. Then it can generate the action potential, right? So stimulus which is not able to generate the nerve impulse is sub-threshold, right? So when to this nerve fiber, when there will be many sub-threshold stimulus, when they will be applied, right? So obviously it will be having an add-on effect, additive effect, right? The summation will be there. Then the summation will be there. So many small together, they are forming that. The value will be above threshold, and that's what we are going to get. Okay, that's what we are going to get. Okay, done. Take care. So then there is all or none principle, right? All or none principle means either yes or no. There's nothing in between. So all or none means when you are talking about the nerve impulse, let's say you have the stimulus, it is above threshold. It is going to generate the nerve impulse here. So it doesn't matter how strong the stimulus is, right? But that will be of same intensity. Let's say if the stimulus is above threshold, definitely the action potential will be generated, and it doesn't matter that stimulus is very strong, then there will be more action potential. No, it has to be above threshold, then the action potential will be generated, right? It has to be above threshold, and then the action potential will be generated. It is like that, or it is like that. Neuron is conducting or it is not conducting. It's not something in between. It is not something in between, right? Either it is conducting the nerve impulse or it is not conducting. There is nothing like something in between. Okay, bache? Right? So size of the stimulus will be same no matter how strong the stimulus might come. Okay? Right? So Ionic theory of nerve impulse can explain this generation. Even then we will explain the generation and conduction of nerve impulse. So Ionic theory of nerve impulse conduction. Ionic theory of nerve impulse conduction. Understood? It is the Ionic theory of nerve impulse conduction. Right? Yeah, Ionic theory of nerve impulse, that's what you can say. Ionic theory of nerve impulse, that's what you can say. So Hawking, right? Who gave it? Hawking. He explained it. Okay, who explained it? Mr. Hawking. Okay, Mr. Hawking. Okay. So it was put forward by English neurophysiologist Hawking, and yes, not just the Hawking and Huxley. Somewhere in 1930, okay, they put forward this theory. Okay, bache? Okay. Right? Okay. So now basically it has to explain that how a nerve cell is getting excited. So when you talk about the excitable nature of this neuron, it depends upon two things. Differential permeability. It depends upon two things. Differential permeability. I'll explain it. And second is your electric. Yes, bache. One is your differential permeability. Second is your electric field. And that electric field across the membrane. These two things are going to decide. Right? These two things are responsible for the nerve impulse, right, generation. What is it? Differential permeability means different permeability. For what? For sodium and potassium ions. Yes, bache. If you want to understand this, right? If you want to understand this topic, yes, you have to remember that when you talk about the sodium ion and the potassium ion, when you talk about the sodium ions or the potassium ions, what is it? Sodium ions and the potassium ions. When we discuss about them, membrane is having different permeability for that. Just let's say when a membrane is resting, that's a very important topic. Please pay attention. I'll write this line also, but here my main agenda is to explain the topic. So when a membrane is resting, when it is in a resting state, so membrane permeability will be more for potassium. The potassium movement, right? Its permeability is more, right? So when the action potential is there, obviously its permeability will shift to the sodium, even that is for some time, but it will be. So these two things are responsible, right, bache? These two things are basically responsible for what? For that electrical events in the nerve fiber. They are responsible for that electrical events in the nerve fiber. Okay. Okay. Doubt? Hanji, any doubt? Done? So these two factors, differential permeability and electric field will decide this, right? It will excite the fiber. Okay. Okay. Done? Chalo. So now I'll explain this. Let's say when a neuron is in resting state. When a neuron is in resting state, then I'll talk about the ion channels and everything also, right? When a neuron is in resting state, imagine the thing. Neuron is a cell, in its surrounding also. Let's take the functional part of neuron, axon. Let's take the functional part of neuron, that is axon. Okay, what is that? It is a functional part of neuron, and what are we calling it? We are calling it as axon. So when you talk about the neuron which is in the resting state, overall you guys can imagine, right? It's a cell. Obviously inside you are going to see some ions, outside also you are going to see some ions. Overall, if you will ask me now, overall, like I'm not saying the total number of sodium ions in and out they are same, or potassium in and out they are same. Normally the condition is that this concentration in between the intracellular fluid and the extracellular fluid is same. Overall plus minus is same there. What am I saying? That when the neuron is in a resting state, plasma membrane is separating two solutions, right, bache? So they are having total number of, right? They have approximately same total number of ions, right? Let's say it is the neuron, intracellular thing. You can say that this is the exoplasm, this is the extracellular. We know that membrane is present in between. So normally, if you will ask me the total number of ions inside and outside, it will be same. If you ask me, Surbhi bache, total number of ions inside and outside, it is same, right? Total number of ions right? It is same. Is that clear? Is that clear? Is that clear? Right. So both the extracellular fluid, even the intracellular fluid, within the axon, total number of ion is same. Total number of ion is same. Right, bache? What am I saying? Overall, right? What am I saying? Ki total number, if you ask me total number of ions, same. It's like if I have to give you the example, ki you have 100 rupees, even I have 100 rupees, like, but you have more coins. I don't have coins. I have that 100 rupee ka note. You have the coins, but ultimately you also have 100 rupees. Even I do have that. You can consider it in this way. But overall, this is something that you need to keep in your mind, that yes, that's a case, right? Obviously, this is something that you need to keep in your mind, that this case is, right, that it is very simple. It is very simple that total number of ions, they are same. But when you talk about the extracellular fluid, when you talk about what? When you talk about the extracellular fluid and within the axon, right? So you will see that outside pe, you will see, sodium ion, even the chloride, they predominates, means their quantity is more. Again, I am explaining it. Again, I am explaining it, guys. Again, I am explaining it. I am saying that sodium and chloride, they predominates. Chloride is playing no role in this conduction and thing. Okay? But chloride is there. It doesn't mean that potassium is not there. Potassium is also there. Potassium is also there. In the extracellular fluid, sodium, potassium, all other ions are present. But what am I saying? That sodium predominates. Sodium's quantity is more. Even chloride is more. But chloride is playing no role, right? But chloride is playing no role in the conduction here. Are you getting my point? Chloride is playing no role in the conduction here. Are you getting my point? Now when it is the axon, so within the axon also, within this axon, the point here is, here within this, right, you people will see that it is the potassium that predominates, means the concentration of potassium is more, sodium is also there, it is less. Plus you will see some negatively charged, organic proteins. Now try to understand it. This is the membrane. Outside I am saying sodium is more and potassium is less. Potassium is more, sodium is less. I'm not saying it is absent. Even chloride is more here. And here I'm saying negatively charged. Organic proteins and negatively charged proteins are present. Now try to understand. Ultimately, we will see the ion movement across the membrane. If ion movement is there across the membrane, then definitely ion channels has to be there. If ion movement is there, right? If ion movement is there across the membrane, it means obviously ions cannot cross directly. The ion channels should be there. Obviously, we need to discuss that. We are literally going to discuss the movement of potassium, movement of sodium, right? And there are different ion channels, pumps that are going to help it. One thing is there, right, that when the neuron is in the resting state, that's what I said, when the neuron is in the resting state, inside is negative, outside is positive, and that is what we have to learn, that how this thing is achieved. Okay, I got this, that when there is the stimulus, electrical disturbance is there, ions will come inside. I got this point, right? I got this point completely, कि ions will come in, that is the point. Okay, fair enough, right? Fair enough. But my point here is, right, very simple. That how can we maintain this thing? Right, So ultimately ion movement is there because total number of ion is same, but still the conditions are in such a way that outside will be positive and inside is negative, and that's what we need to know how. So definitely there are ion channels which are present here on the membrane, right? And that's what we need to discuss now. Jaise ye dekho, right? Sodium potassium pumps, will come to this, right? Sodium potassium pumps, and see this diagram. Look at the ion channels here. This is the extracellular fluid. Sodium is more here. Intracellular fluid, potassium is more here. You have the leakage channels. You have the voltage-gated ion channels, ligand-gated ion channels, signal-gated ion channels. There are different types of ion channels. Are you getting my point? 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 Yes, please. Yes, please. Any doubt here? Any doubt here? Pakka? Sure? Sure? Firstly, understand this. Then I will start. I'll tell you how the things are going to start. So obviously ion channels are there for sure. Ion channels are there for sure. Right? There are channels, and we understand the plasma membrane's structure. So yes, we know how the things are. Right? Right? So you guys can see this, right? So there are different types of ion channels that we have. Okay, this is the phospholipid bilayer. As I said, leakage channels or leaky channels, voltage gated channels, ligand gated channels, signal gated channels. Mainly we are going to talk about these leaky channels. We will talk about voltage gated channels. We will even discuss about sodium potassium ATPase pump. We are also going to discuss about sodium potassium ATPase pump. Right? So we will talk about the leaky channels. We will talk about the voltage gated ion channels. We will also talk about the sodium potassium. Right? We will also talk about the sodium potassium ATPase pump. Okay? Okay, ready, right? So there are different types of channels. What do we have? There are different types of channels. Is that clear? Is that clear? Is that clear? Yes, yes. Chalo, let's discuss it, right? So overall, and it's not about the neurons. जैसे देखो, some channels mechanical channels. जैसे when we apply the pressure, we can feel it. Somebody is holding our hand like this, we can feel it. Pain, we can feel. Pain receptors are there, right? Like somebody is exerting something, some pressure, we can feel it. Mechanical, right? Right, bache? Mechanical. Yes, bache. Mechanical. Are you getting my point? What do we have there? What do we have there? Mechanical gated channel. So I am writing the types of ion channels here, and you should know this. I am writing the types of ion channels here, and you people should know it. Right, bache? So as I said, even in the diagram it is given that there will be leakage channels. Yeah, leaky channels are there. Leakage channels, I'll explain it. Right. Voltage gated ion channels. Voltage gated ion channels, and we know that when it comes to the nerve impulse, right? Nerve impulse, conduction, nerve impulse, this nerve impulse, right? And its conduction, we'll talk about the sodium and potassium ions, their differential permeability. So we will obviously, our discussion will be more around that. So mechanical gated ion channels, and we will have ligand gated also, right? Matlab here it will be because of the change in the voltage. Ligand gated matlab that particular sodium or something will join with it, right? That's what we need to discuss. And we have the pump. We have the pump. So bache, if I'm using the pump, if I'm using the word pump, then what is it? If I'm using the word pump, matlab these are the ion channels that we discuss. We'll also use this word pump. So pump, sodium, potassium, ATPase pump. We will also talk about it. Sodium, potassium, ATPase pump. So if it is a pump, ATPase pump, so it means it will help in the movement of things against the concentration gradient. Remember, against the concentration gradient, A for ATP will be used. Against the concentration gradient, ions will move, and A for ATP will be used. Right? A for ATP will be used. That's what we need to discuss. That's what we need to discuss. So when you are talking about the leakage channels, you have sodium leaky channels or leakage channels, and potassium leaky channels or leakage channels are there. We'll explain it, what is the meaning of that. Voltage gated ion channels, again sodium voltage gated ion channel, potassium voltage gated ion channels. So we are going to talk about it mainly, right? We are going to talk about it mainly. Is that clear? And then we'll talk about the pumps. Okay, okay. Yes, bache. Yes, bache. Yes or no? Yes or no? Right? So the word is, so as I said, we need to focus on these things. Okay? We'll discuss that these things only. Voltage gated ion channels, leakage channels, sodium, potassium, ATPase pump. They are going to help, right? They are basically involved in this. Ionic, their nerve impulse, the excitability of neuron cell, and then the conductivity of that, right? They are involved in that. Okay, so firstly, let's talk about these leaky channels or leakage channels. Yeah, focus on this one, leaky or leakage. Leaky or leakage, just as simple it is. Let's consider this is your neuron. It doesn't look like, but still, let's say this is your axon, this is your neuron. Ion channels are there. Why? Because for the movement, right? For the movement, right? Ion channels are there. Why are they present here? For the movement, of course, right? For the movement, of course, 110%. 110%. Now, it is the leakage is the word. Randomly things are moving. Don't you think so? Leakage. Let's say there is a box, there is a container, not airtight, right, not properly closed, not airtight. Obviously, things can just get out, come out. They can just come out. You can understand this word now. So as I said, when you are talking about the leakage channels, we have sodium leakage channels also. Yeah, leaky channels word you can also use, and potassium leakage channels also. Potassium leakage channels are also there. And bache, they will, how the things will move across it. down the concentration gradient. मतलब randomly these channels will open, randomly these channels will close, and down the concentration gradient things will move. Down the concentration gradient things will move. How the things will move across it? Down the concentration gradient. Down the concentration gradient. मतलब along the concentration gradient. And randomly these channels, right? Randomly they open or close. They open or close. Now, I told you when it comes to the electrical excitability, the differential permeability, I told you when it comes to the electrical excitability, differential permeability and the electric field across the nerve fiber is going to decide that. Yes or no? Yes or no? Yes or no? है ना? It is going to decide that. So I told you that there will be the differential permeability for sodium ion, for potassium ion, and that too in resting state or during the action potential. So what I just said here, that down the concentration gradient, the movement will be there, randomly they open or close. Now remember, your neurons, they have more sodium leaky channels, more potassium leaky channels, then I'm writing it correctly, but chalo, have more potassium leaky channels than sodium leakage channels. They have more potassium leakage channels than sodium leakage channels. That's what I just said. That is what I just said. Okay. Dekho, for an example, this is your axon. Yeah, you can say that this is your intracellular fluid. This is your extracellular fluid. Just imagine it like this. I told you this is the condition. Outside you have more sodium and less potassium. Inside, this point is important, students. Inside you have less sodium, more potassium. Now you tell me, right? Now you tell me this thing. Please, you tell me this thing. If the things will go, right? If the things will go as per or down the concentration gradient. So you tell me what will be the movement of this potassium ion. The potassium ion will move from inside to outside or from outside to inside, right? What will be the movement of potassium ion? Tell me. Yes. Yes. But what I just said, I'm talking about the leakage channels. I'm talking about the potassium. I'm talking about the potassium and the sodium leakage channels. I am saying, right, that let's say leakage channels are there, right? So here the movement will be down the concentration gradient, down the electrochemical gradient. So we know that sodium is more outside. Sodium is less inside. Don't you think this is the outside, sodium is more, potassium is less. So obviously sodium from outside will come inside. From outside will come inside. है ना? And inside you have more potassium. So potassium will go outside. So down the concentration gradient, that is the movement. That is the flow of the movement. Please try to understand it. Down the concentration gradient, that is how the things are going to be. So when you talk about the leakage channels, right? So from these leakage channels, we know that sodium is more outside, sodium is less inside. So sodium from outside will come inside within the neuron, within the axon, and the potassium, which is more inside, right, that will definitely go outside. Now we say one line, that resting nerve fiber, we say this thing, very important thing, that resting nerve fiber is more permeable. I think I have repeated it multiple times. Resting nerve fiber is more permeable to K positive than Na positive, right? And the reason for that is, you can also take it in this way, that neurons are having more K positive leakage channels than the but they have more K positive leakage channels than the Na positive leakage channels. So obviously more, yes, obviously what's going to happen? What is going to happen? Please tell me. What is going to happen? More sodium will come out? Yes or no? Yes, please. Yes, please. Got it? Yes. Okay? Thank you. So this is what you need to keep in your mind. This is the one thing, right? This is about the leakage channels. Now, obviously, I told you outside sodium is more, even the chloride is predominant. And I told you that inside you will have more potassium, less sodium, and even negatively charged, right? Even negatively charged proteins. Bache, proteins, they are biomacromolecules. If you think that these proteins are going to leave, right? If you think that these proteins are going to leave the exon here, obviously no. So because already we have less, see, take it in this way. I am saying inside you have potassium more, you have less sodium, plus negatively charged proteins are predominant here. And I told you about the leakage channels, and I am saying outside your sodium is more and outside your potassium is less. Now my point here is, right, we have more sodium here, right, but still sodium will tend to diffuse. It will tend to diffuse because of these leakage channels thing, right? And in the comparison to that, less sodium will enter. Why? Because sodium leakage channels are less in comparison to the potassium leakage channel. Second thing, bache, these negatively charged proteins, they will not come outside. So inside, obviously the negative charge is more, right? The positive charge, whatever positive charge is there inside, that is also coming out, right? And less sodium is entering here. So obviously even the negatively charged proteins are there. So definitely inside will be more negative. Is that clear? Inside will be more negative. Is that clear? Right? So this differential flow of positively charged ions and inability of negatively charged ion within the fiber to pass out cause an increased positive charge on outside and negative charge on the inside, right? So that's why the nerve fiber is polarized. That's why inside it is negative and outside it is positive, because chloride ions are to playing no role. I told you that, right? Yes, that's what I'm saying now. So because of this membrane differential permeability, because it is sending more potassium outside, comparatively less sodium is coming in because it is more permeable. In resting state, it is more permeable for potassium and negatively charged protein is there. So that's why inside is becoming negative, outside is becoming positive. But remember, if the negative charge inside will keep on increasing, the potassium exit will also slow down. This is one of the reasons. Second reason is your sodium potassium ATPase pump. Right now I'm telling you why membrane is in polarized state at rest. Sodium potassium ATPase pump. As I said, it's a pump. It is a pump. What's that, guys? It is a pump. So from this pump, yes, a sodium potassium ATPase pump. So here you have two tricks. One is Nokia and Kintu. Two tricks are there. Nokia, Kintu. You have two tricks here. Nokia, Kintu. Nokia phones, I hope you know about it. N stands for sodium. O stands for out. K stands for potassium. I stands for in. A stands for ATP. A stands for against. Are you getting my point? There are two tricks that you need to remember here. Nokia, Kintu. right? These are two tricks that you need to keep in your mind, and that is Nokia Kintu, Nokia Kintu, Nokia Kintu. Getting it? Yes. So Nokia means N stands for sodium here, O outside, means with the help of this pump, sodium is moving outside, right? Sodium is moving outside, yes or no? And potassium is coming inside. How? With the use of ATP. That is against the concentration gradient. Already you know that sodium is more. Sodium is, sorry, my bad. Kintu na, huh? Yes, it's right. So you know that, already you know that ki sodium is more outside, right? And potassium is more inside. And whatever sodium is coming here, it is less in comparison to the potassium diffusing. So it is Nokia Kintu. Matlab sodium will go outside from. Sodium will go outside. So 3 sodium ions will go outside and 2 potassium ions will in. So what is the meaning of this Kintu? K means potassium, of course. K means potassium oxide. It's Kintu. I means in. Means your potassium is coming in. Right? If you ever get confused, it's Kintu. Kintu means 2 potassiums are coming in and 3 sodiums are going out. 2 potassiums are coming in, 3 sodiums are going out. 2 potassiums are coming in, 3 sodiums are going out. So in sodium potassium, ATP is pumped by the use of ATP against the concentration gradient. Right? Obviously it is against the concentration gradient. Obviously inside we know that less sodium is there, but still the sodium is from inside is going outside. So 3Na positive is going outside, and in comparison to that 2K positive, they are moving inside. So obviously that is also responsible for having inner side as negative and outer side as positive. That is also the reason that the membrane is in polarized state. That is also the reason that the membrane is in polarized state. Am I right or wrong? Am I right or wrong? So normal difference in the ionic concentration and electric potential between outside and inside, it is also maintained by this active transport. Okay, it is also maintained by this active transport. Got it? Yes. Got it? Survey, it has to be in this form now. I told you differential membrane permeability, inside negative, outside positive, and this electric field across, that potential difference is deciding the things. So that is the case when the nerve impulse, right, when the neuron is not conducting any nerve impulse. Let's say now it is getting a stimulus, right, obviously above threshold. What's going to happen when there is a disturbance? I'll tell you, I'll explain. See, that's the neuron. I told you that this is the most sensitive part, axon hillock. That's the most sensitive part, axon hillock. Right? Why? Because it is having more number of ions. So when a threshold stimulus will come, obviously the ionic disturbance will be there. So sodium will start moving inside them at the point of stimulus. It's not like that immediately, it's not like that immediately ions will start moving. No, at one point the sodium, right, the sodium will start moving inside. The sodium will start moving inside, right, bache, at the point of disturbance, right, the sodium will start moving inside. So whenever the neuron will receive a nerve impulse, whenever it will generate nerve impulse, the sodium influx will be there. The sodium movement will be towards the inner side. This point clear? This point clear? And look at this sodium potassium ATP is pumped. So with the help of ATP, it will throw 3 Na positive outside and 2 K positive inside. So that is why the trick is Nokia Kintu. That is why the trick is Nokia Kintu. Got it? That is why the trick is Nokia or Kintu. Got it? Yes? Yes? Hanji Surbhi. Okay, chalo. So, looking for the better pictures. Chaliye, it's okay. So that's why it is like that. So let's say this is the point where it is receiving. So I told you that at that axon hillock, most sensitive part. So that's how it will come in. Chalo, theek hai, okay. So sodium will start moving in. Sodium influx will be there. Now when sodium will start moving in, right? When sodium will start moving in, obviously it is going to change the ionic concentration. It is going to change the ionic concentration within, right? Inside will start becoming positive, outside will start becoming negative. But how is it possible? Because of the sodium influx, right? And I'll tell you what factors are going to support it. I'll tell you what factors are going to support it. But firstly, tell me, are you able to understand this? Are you able to understand this? Yes? 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 So students, I am Anji, so this point is clear, right? I believe it is clear. Yes? Sure? Please tell me, is it clear? Okay. Chale. So now the next thing that we need to understand here is that there is the entry of sodium within. Now I told you about the, yes bache, I told you about what? I told you about this thing now. What do we say? I told you about the ion channels. I told you about the voltage gated ion channels. If you remember, when I was telling you about the ion channels, I told you about the things. Voltage gated, you know about the leakage channels, but we have voltage gated sodium channels and we have voltage gated potassium channels as well. That's what we have. Voltage gated sodium channels and voltage gated potassium channels. Now the word is voltage gated. Now the word is voltage gated. Voltage means they will open or close at a change, at a specific change, yeah, at a change in the voltage, right? So how that voltage will change? When the membrane permeability will change, when the ion movement, because of this ion movement. For an example, when there is a disturbance, right? Let's say this is the point A where there is a, when it is receiving the stimulus, means there will be an influx of sodium ion. Your sodium ion will come in. Your sodium ion will start moving in, right? So if sodium ion is start moving in, means more positive is coming inside. Don't you think it can change the membrane polarity? Don't you think it can change the membrane polarity? Definitely. Don't you think it can change that membrane, that potential difference? Obviously. Don't you think that can then open the voltage gated sodium channels? What do you think? What do you think? You tell me and tell. You please think about it and tell me. You please think about it and tell me. Yes. Tell me. You know now, action potential. We are basically, now we will understand that how this action potential will be generated. So action potential means depolarization has to be done. And yes, we will also discuss about the hyperpolarization. We'll also discuss about the hyperpolarization. That is also a word. But mainly what am I saying? That depolarization has to be there. Depolarization has to be there. Right? So if depolarization has to be there, so it means, right, what is the meaning of that? That ion movement, right? That negative membrane potential, right? Matlab inside should be positive, outside should be negative. That's what we have to do, right? Inside should be positive, outside should be negative. That's what we have to do. Yes or no? Yes or no? Yes or no? है ना? So what's going to happen, bache? What is going to happen? That we know that, right? We know that inside, like when the neuron, when it was resting, we know that inside was negative, outside was positive, right? So obviously ion movement has to come in. So to generate the action potential, firstly this negative has to reach to 0, right? Just let's say if it is like this, here you have negative, this is 0, here you have positive. Here you have positive, right बच्चे? Right बच्चे, जैसे -1, -2, -3, +1, +2, +3, and we know that when the membrane is at rest, right, when it is the resting potential, inside is negative, so that negative first has to reach to 0, then it has to go to positive, then it has to go to positive, it has to reach 0, then it has to go positive, and when there is the action potential, remember, inside will be positive, somewhere around +20 millivolt, right, somewhere around +20 millivolt. Is that clear? Is that clear? Right, so the origin and maintenance of resting potential depends upon, I told you, it depends upon the disturbance caused, right, it depends upon, on the leakage channel and sodium potassium ATPase channel. ठीक है बच्चे? So now we have to decrease this transmembrane potential, right बच्चे? So membrane, now what's going to happen? Because there is a stimulus, membrane's polarity will change, permeability will change. Because of this stimulus, because of this disturbance, membrane's permeability will change. What's going to happen? Membrane's permeability will change. Now what's going to happen? Right, I told you, now when there will be the action potential, membrane differential permeability is the reason for the nerve impulse conduction. So what's going to happen? Now it will become more permeable. Now it will become more permeable to sodium than potassium. Right, and that is for few seconds only. It's not like that it will this phase will last longer. Of course not. Of course not. Right, of course not. Right, of course not. So here what's going to happen? Here what's going to happen? When obviously it will receive the stimulus, the membrane's permeability will change again. Now it will become more permeable to sodium than potassium. So if it is more permeable to sodium than potassium, it means more sodium will come in. Now if more sodium will come in, this negative will reach to the positive, and that will result in the opening of your voltage gated sodium channels. That will result in the opening of your voltage gated sodium channels. And bache, if voltage gated sodium channels will open, means more sodium will come inside, means at here more plus charge will come, outside will be negative, means the membrane will become, at this point where it has received the stimulus, it will become depolarized, means action potential will generate. It will become depolarized. It will be there for some time, but it will become depolarized. Now this depolarization will move from this part to this part to this part to this, across the length of the axon. Right, down the axon. That's how it has to move. It's not like that just in one go, immediately it will become reverse, inside positive, outside negative. No. At the point of stimulus, membrane's permeability, right, slowly it will change. It will be more permeable to sodium than potassium. More sodium ion will come in, that results in the opening of the voltage gated ion channels. Right, bache? Right, bache? Right, so this property, because membrane potential is decreasing, membrane is becoming more permeable to sodium. So due to the influx, sodium is coming, membrane's permeability is changing, it is becoming, because from minus it will reach to 0 and to positive, it has to reach there. So when there is a change in that potential difference, its permeability is also changing, and that is the thing which is making this neuron or neuron membrane unique. Right, that is why it is not just like the other one. Just like the other one, right bache? Right bache? Okay, so as the transmembrane potential decreases, okay, so what's going to happen? Membrane will become more permeable to sodium than to potassium. Right? 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 So now your voltage gated ion channels will open, and because of that, what's going to happen? More sodium will come in. Right? Yes? Yes or no? Yes or no? है ना? So obviously, so when there is the wave of action potential, your sodium channel, right, they will open. It is still closed. They are open and it is still closed. So because of that, more sodium will come in. Inside will become positive, outside will become negative. Inside will become positive, outside will become negative. That's how it is. So these channels, they will only open at specific voltage and then they will close by itself. right? Then they will just get closed. They will just get closed, right, bache? So that is happening here. That is happening here, right, bache? So you can see that these gated channels, they are peculiar in being self-closing. Okay, okay. Right? So these sodium and ion channels, you know that they pass by a different or separate and independently controlled channel. So when there is the resting potential, right, during resting potential, both these channels are closed. But when there is the action potential, firstly, this channel will get open. Is that clear? Is that clear? And there will be this depolarization. Now come to this diagram. जैसे ये देखो, sodium influx is there when it will receive stimulus. So obviously inside the transmembrane potential will become 0 and then become positive, and that will result in the opening of voltage gated ion channel. So obviously more sodium will come. So when more sodium will come, inside will become positive, outside is negative. So this is your depolarization, right? So it's just like I told you, electric current around the membrane and the membrane differential permeability will help in the generation here. So obviously it's positive, it's negative. At this point, inside is positive, outside is negative. This plus will move here, this will move here. Means, what does it mean? From A point, your electric impulse will move to B point. So because of that, what's going to happen? Your B will become, right, inside will become positive, outside will become negative, right? That's how it is now. Current is moving from plus to minus, right? Right, so inside is positive, outside is negative. Here, inside will become positive, outside will become negative. Then again here, throughout the length of axon, that's how it is going to move. It's going to move. That's how it is going to move, right? Right. So remember one thing, no doubt, membrane will change its permeability. Membrane will change its permeability, right, bache? Right, entry of sodium will result in depolarization. Nerve fiber will become electropositive with respect to extracellular fluid. Right, but this reversal of polarity is sudden. This reversal of polarity is sudden and momentary, right? It will just reverse immediately after that. It is sudden and momentary, and it will last for just few seconds, 1/1000th part of the second, like this. Then again it will reach back to repolarization. So there are three things that we discuss. Inside negative, outside positive. We are calling it as polarization, right? And you know that your leaky channels, right? Leaky channels and sodium potassium ATPase pump is responsible for that. Then the status of inside negative, inside positive, outside negative. This is depolarization, when the action potential will get generated. So here what's going to happen? Leaky channels are always open, but here what will you see? Voltage gated sodium channels will get open. So obviously this depolarization, for it is momentary. So again the repolarization will occur. Again, the inside will become negative, outside will become positive. This is the repolarization. And it is the time when your sodium, voltage gated sodium channels will open. So now I will make a table here, right? And that table will provide you a better idea of this particular topic. But firstly, tell me the ion movement and everything clear? Ion movement and everything clear? So when there is the depolarization, polarization, when there is the polarization, so we can discuss it in this way, that passive diffusion. When I'm talking about the passive diffusion, bache, I'm talking about that leakage channels, leaky channels, right? You can talk about the voltage gated, sodium voltage gated channel, potassium voltage gated channel, and sodium potassium ATPase pump. And sodium potassium ATPase pump. So obviously when there is the polarization, yes, there will be the passive diffusion. Yes, your voltage gated ion channels, your leakage channels are open. But here no, here no, here yes. Am I right? So when there is the polarization, so passive diffusion, that is your leakage channels, your both leakage channels are open, right? Both leakage channels are open, but your voltage gated channels are closed, right? Sodium potassium pump is open. Now because depolarization is there, so at the time of depolarization, passive diffusion, it's going on, right? Passive diffusion, passively the things are going on. It will not stop. That passive things are going on. These channels randomly open and close, but passive movement is still there, right? But it is the depolarization. We want more plus in. So during that, right, this, your voltage gated, sodium voltage gated channel will be open. This one is closed. Yeah, I think I should write open and close only. Open, closed, closed, open, like it's functional. Passive diffusion again here, working. This one is open, this one is closed, and this one will also be closed here. When there is the depolarization, even your sodium potassium ATPase pump will get closed. Now when there is the repolarization, again we want our membrane. Again we want our membrane to become polarized. So obviously open, but this channel has to close because now we want more potassium outside. So it will be open and this one will also be open. That's the case. That's the case. Is that clear? Is that clear? Is that clear? And the same thing will be there during the hyperpolarization, right? So what is initiating depolarization? The movement of sodium inside. When sodium is more inside, the transmembrane permeability will, the transmembrane potential difference will also change, right? Yeah, you can say the transmembrane potential decreases. Transmembrane potential decreases, inside will become positive, outside will become negative. That will result in the opening of voltage gated ion channels because there is a change in voltage due to which more sodium will come in, right? Inside is positive, outside is negative. Now let's say if stimulus has generated here, so it has to travel down the axon, yeah, along the axon. It has to travel down the axon, yeah, along the axon. That's what you need to keep in your mind. That's what you need to keep in your mind. That's why it has to move. So charge, current, plus to minus, plus to minus. So let's say if your depolarization moved from this point to this point, again here you will see repolarization will occur. Repolarization will occur, right, bache? But let me tell you that when you talk about the neuron, firstly it will carry that nerve impulse to the end. Synaptic vesicle will release neurotransmitter. Then there comes, it will pass the nerve impulse to the next neuron. A refractory period will come and then it will again receive the next stimulus. It's not like that one stimulus is here and it is also receiving next stimulus. No, it will be like, this is my work, right? I have to do this, I'll complete this work and then I'll take the next one, right? No multitasking here. No multitasking here. Okay. No multitasking here. Okay. Okay, sure, right? So repolarization mein, what's going to happen if I have to write it? Yes, what is going to happen, bache? So permeability of the membrane to sodium ion drops, right? 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 So now the permeability of the membrane for sodium ions drop, but still by that time there is more sodium inside. Okay, okay, right? Yes. Yes. Then outside. And because of that, what's going, and yes, parallelly you will also see that membrane. Parallelly you will also see that membrane become more, membrane will become permeable to. At the same time, you will see membrane will become permeable to potassium ion, more permeable to potassium ion than sodium ion. So at the same time, membrane become more permeable to potassium ion than sodium ion. Got it? Potassium ion then sodium ion. Is that clear? Is that clear? So there will be the opening of K positive channel, right? So because of that, there will be the opening of voltage gated K positive channel and voltage gated sodium channels will close. Will close, right? Because repolarization has to achieve, right? Right? So more potassium ion pass out, right? And in comparison to the sodium enter, then again membrane will become. Because of that, what's going to happen? So due to which inside will become negative and outside will become positive. So that is how it works. So it is not difficult. So you have to remember the things that electric field and differential permeability of the membrane is responsible for all this. Number one, during repolarization, voltage gated ion channels, they are open, right? Leakage channels, they are playing a huge role here, right? But during repolarization, sodium potassium ATPase pump is very important for maintaining that repolarization. Sodium potassium ATP, one second. So what you need to remember that during polarization, right, your leakage channels are playing the role. Neurons are having more number of potassium leakage channel than the sodium leakage channel, due to which more potassium will diffuse out in comparison to the sodium that comes in. Second thing is that the sodium potassium ATPase pump, it is present on the neurons membrane. So 3 sodium ions will move outside, 2 potassium ions will come inside. That is also maintaining that inside negative, outside positive condition. And at this point of time, voltage gated ion channels, they are closed. At this point of time, voltage gated ion channels, they are closed. What about the voltage gated ion channels? They are closed. Is that clear? Is that clear? Is that clear? Sure. Voltage gated ion channels, they are closed. Understood? 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 Okay. Then when there is the depolarization, then stimulus will come. Obviously it has to be above threshold value. Then it will come. Because of that, sodium ions will come inside, right? Sodium ions will start coming inside. So transmembrane permeability, like transmembrane potential change will be there. Because of that, voltage gated ion channels, sodium voltage gated ion channels will open. Right now, more sodium will come inside. Inside will be positive, outside is negative. That is depolarization, and that electrical impulse has to pass. That nerve impulse has to pass along the length of the axon. That's the thing you need to keep in your mind. But this change in membrane permeability is for some time only, right? For some time only, the neuron is allowing more sodium to come inside. Yeah, its permeability is more for sodium than the potassium. Then again, there is the reversal of polarity, right? What is going to happen? What is going to happen, right? See, this is going to happen, right? Immediately the membrane permeability will again change. Because of that, what is going to happen? Because already we have more positive inside, yeah, more sodium inside in comparison to outside, right? That will also help in the opening of voltage gated potassium channels, right? More potassium will start moving outside. Now at this point of time, voltage gated sodium channels are closed, right? And because of that, what's going to happen? Inside will become negative, outside will be positive. And already we know that within the neuron, negatively charged organic proteins are present that cannot leave it. So inside is more, right? Inside is already more what? It is more negative. Understood? Understood? So this is all about the generation and conduction of the nerve impulse. Ionic theory is explaining it, and right? It is the generation of the nerve impulse, and it is across the myelinated nerve fiber. It is across the myelinated nerve fiber. Right, bache? When it will be the, already I told you, when it is the, sorry, it is against the non-myelinated nerve fiber. So when there is the myelinated nerve fiber, right? So the nerve impulse will jump from this point to this, then this to this. So obviously it will be faster. It will be faster, right, bache? And we call it as saltatory conduction. We call it as saltatory conduction. Got it? We call it as saltatory conduction. Got it? ठीक है, चलिए. So now you know about the neuron endings. जैसे let's say this is your one neuron. You know that axon, right? You know what axons are going to do. You know that, na? What axons are going to do. Obviously, what is going to happen? That nerve impulse will reach here. Nerve endings are having synaptical vesicles, and these vesicles are loaded with what? These vesicles are loaded with neurotransmitters, right? They are loaded with neurotransmitters. And now you know that because here the point-to-point connection is this. So obviously you have one more neuron just next to it. You have one more neuron, and this one is going to receive the impulse, and then it will pass it further. We know it. Now we are going to focus on this particular part, my dear students, right? Just let's see. This is the neuron ending, axon ending. This is the synaptical vesicle. Here you have the neurotransmitter. Now, bache, when the nerve impulse will reach here at the end, some changes will occur. We will discuss that. But you understand one thing, right? See, this is your one neuron, this is your another. This neuron, and there is a gap in between. This gap is known as synaptic cleft, right? This is your one neuron, this is your another neuron. There is a gap in between. We call it as synaptic cleft. We call it as synaptic cleft. And this is presynaptic membrane. This is presynaptic membrane, right? And this is postsynaptic membrane because it is after the synaptic cleft. Okay, so it is basically the axon ending. It is basically the dendrite, right? You know that axon is carrying the nerve impulse away from the cell body. Dendrites will carry it towards the cell body. So firstly, tell me this portion clear. Firstly, tell me this portion clear. Yes, please tell me this portion clear. Yes or no? Yes or no? Yes or no? Right. So right now what are we discussing? We are discussing a synapse, right? So presynaptic membrane or presynaptic node, synaptic cleft, postsynaptic membrane. These three things are making your synapse. These three things are making your synapse, and it is the chemical synapse, because here we will talk about the release of this chemical neurotransmitter. So this is the chemical synapse. We have electrical synapse also. We will talk about it. Right, so what's that? It is the chemical synapse. What's that? It is the chemical synapse. Is that clear? Is that clear? Is that clear? Okay, so I believe this portion is clear. I believe this portion is clear. Right, these are the gaps that are present. We'll see how that nerve impulse will pass from this neuron to this neuron. But tell me, this gap part clear? So Sherrington used the term synapse for the first time. Right, Sherrington used the term synapse for the first time. Right, first use the term synapse. Okay, okay, yes. Tell me. So firstly tell me, this part clear? Sure? Yes? Okay, so I am talking about the neurotransmitter here. So we will take the example of acetylcholine. But we have two types of neurotransmitters. We have, when you talk about the neurotransmitters, na? When you talk about the neurotransmitters, when you talk about the neurotransmitters, we have two types, excitatory, inhibitory. Matlab one which is sending, right, which is basically excitatory or inhibit. Inhibit means to stop, excitatory also neurotransmitter, which will tell, this is, let's say when we talk about some drugs, they slow down our system, they make us active. Same is the case here. Same is the case here. Some are going to stop it, right? Some are going to promote it. Are you getting my point? Some are going to promote it. So excitatory and inhibitory, these are the two types of neurotransmitters. These are the two types of neurotransmitters. Is that clear? So now in your NCERT, yes, what's the word here, bache? We have excitatory and inhibitory neurotransmitters. So when you are talking about the excitatory, right, the acetylcholine, that is given in your NCERT, that is the example of your excitatory neurotransmitter. Yes or no? Yes or no? Epinephrine, also known as adrenaline, your norepinephrine, that is also the example of your excitatory neurotransmitter. Right, bache? Right. Then you have GABA. What is GABA? Gamma amino butyric acid. That is the example of inhibitory. You have glycine. That's an amino acid, inhibitory. Even the serotonin is also considered as the inhibitory neurotransmitter. Right? They are also considered as the inhibitory neurotransmitter. Is that clear? Is that clear? Is that clear? So these are the chemical messengers, bache. They'll carry our message from one nerve cell to the next, even to the muscle or to the gland they can pass. Okay? Okay? Done? Please tell me, is it done? So excitatory done, inhibitory done, right? So examples should be clear to you. Examples should be clear to you. So these are the neurotransmitters. Now excitatory. Let's take the example of acetylcholine. Let's take the example of acetylcholine. Let me tell you these your electrical impulse, your nerve impulse will reach here at the end. Now when your nerve impulse is reaching here, obviously membrane polarity is reversed now, inside positive, outside negative, inside positive, outside negative. But due to which this part will allow the calcium ions to come in. Means here the calcium channels will become like this. Your calcium channels will be open here. Okay, ji? So when the impulse arrives at the axon terminal, the synaptical vesicle will move towards the membrane. Why, bache? Why? Why is it so? Please tell me. Because it is becoming, this membrane is becoming permeable for calcium ion. So calcium ions, dekho, calcium ions will enter here. These synaptical vesicles will move to this part, right? And here they will just get fused. And obviously because of that, your chemical messenger, neurotransmitter will release here, right? Now to the next neuron here, you will see some receptors are present. I'm saying na dendrite, this is the membrane, this is the dendrite of next neuron, this is the postsynaptic membrane. Here also you have some receptors. Here also you have some receptors. So what's going to happen when your nerve impulse reaches here at the end, because there is a change in the membrane permeability here, obviously calcium ion, this membrane will become more permeable to calcium ion, and that will help these vesicles, synaptical vesicle, to fuse with plasma membrane, and the neurotransmitter, just your acetylcholine will be released here. Right, bache? Right, bache? Yes? Is that clear? Yes? Yes. Okay. So now because of that, because this chemical messenger will bind to the specific receptor. This chemical messenger, which is released here in synaptic cleft, it will bind to a specific receptor here. It is going to bind to a specific receptor here on next neuron. Because of that, again, the ion channels will open, and this neuron will also generate the nerve impulse. This neuron will also generate the nerve impulse. So now when you talk about this neurotransmitter, because it has to bind with the specific receptor, because of that the membrane permeability will change. Here also the nerve impulse in the next neuron will generate. Right? right? That is the point. So here in the synaptic cleft, just say we have the acetylcholine. So you will have the enzymes that will break these neurotransmitters as well, right? Just say acetylcholine esterase, acetylcholine esterase. Just say acetylcholine is the neurotransmitter. Your acetylcholine esterase enzyme is also present. This enzyme will break your acetylcholine into acetyl and choline. Matlab this, it's not like that this neurotransmitter will stay here only. No, it will release here. It will bind to the specific receptor. Next neuron, in the next neuron, the permeability will change. The impulse will pass to the next neuron. And what is going to happen here? Right, this neurotransmitter after some time, like, or it's not like that it's a very, it will, neurotransmitter will stay here for a longer time. No, it will just get destroyed. It will just get destroyed because of this acetylcholine esterase. So acetylcholine esterase will break it. Acetyl plus choline will form, and this neuron will again, you can say resequester it, will again take it, and again within it, again it will form the neurotransmitter here. So it's not like that ki acetylcholine is there, the enzyme will break it and it is of no use for us. No, the presynaptic neuron will be like, give it back to me. I'll again in my synaptic vesicle, again I will form this neurotransmitter. It's like that. Okay, bache, it's like that. That's how it works, right? That's how it works. Done, bache? Done, bache? Yes. Right? So it is an excitatory neurotransmitter, so the nerve impulse will pass to the next neuron. But if it will be the inhibitory neurotransmitter, it will not let the nerve impulse to pass. The blockage will be there. Why? Because chloride ion channels will open that will block it. Okay, so jaise bache, some people, they take antidepressants, right? They take antidepressants, Prozac or something. What will be the function of that antidepressant? Just say, let's say serotonin, happy hormone. Serotonin is considered as happy hormone, right? Yeah, whatever neurotransmitters are there, like that, some antidepressants, they work like that. They will not let these neurotransmitters to get degenerated very quickly, to break very quickly. They'll let them stay, right? So that further, conduction can be done like this. Okay. That's how it works. Fine. That is how it works. Done. So ye dekho, transmission of nerve impulse. So synapse is there. They are explaining it here that a nerve impulse is transmitted from one neuron to another through the junctions known as synapses. So synapse is formed by the presynaptic neuron, membrane of presynaptic neuron, postsynaptic neuron, which may not be separated by a gap called synaptic cleft. So these three things will form synapse. So as I said, two type of synapses there: electrical synapses, chemical synapses. So electrical, rare, right? So membranes of pre and post synaptic neurons, they are in close proximity. So electric current will flow directly from one neuron to another, right? So it is just like, so transmission of an impulse across electrical synapse is very similar to impulse conduction along a single axon, right, bache? So obviously electrical impulse is always faster than that across a chemical synapse, but it is very rare in our system. It's like this. So yes, let's say this is your presynaptic neuron, this is your postsynaptic neuron, this is the ion channel. So obviously immediately it will pass. There is no synaptic cleft or something. It will directly pass like this. But it is very rare in our system. It is rare in our system, right? We have the chemical synapse. Okay, right? So at a chemical synapse, you people can see the membrane. The membranes of the pre and post synaptic neurons are separated by a fluid-filled space. So your synaptic cleft, right? Your synaptic cleft is fluid-filled. Okay. So neurotransmitters are involved in the transmission of impulse. So axon terminal, they are having vesicles filled with these neurotransmitter. So when action potential arrives at the axon terminal, it is going to stimulate the movement of synaptic vesicle. Why? Because calcium permeability will increase. That will help the synaptic vesicle to get fused and the neurotransmitter will be released in the synaptic cleft. That's the diagram. Look at the receptors here, right? So presynaptic membrane, synaptic cleft, postsynaptic membrane, they are forming the synapse. Okay, they are forming the synapse, and they are forming the synapse. ठीक है बच्चे? So the released neurotransmitter will bind to their specific receptor that is on the postsynaptic membrane. Ion channels will open, entry of ion channels will be there, and a new potential in the postsynaptic neuron will generate. It can be excitatory or inhibitory. Okay, excitatory or inhibitory. So there is the refractory period also, bache. There is the refractory period also, right? This is the period of inexcitability. Okay, what is it? It is the period of inexcitability. Okay, bache. Okay, bache. Right? Okay, so refractory period, so it is a period of excitability during which the stimulus cannot produce a second impulse, right? So the propagation of each impulse is followed by a period of, yes bache. So after the propagation of this each impulse, right, it is followed by a period of inexcitability. It is followed by a period of inexcitability. Are you getting my point? And during which the stimulus cannot produce. During which stimulus cannot produce a new impulse, yeah, second impulse. Right, that is the refractory period. Okay, bache. So in this duration, your nerve fiber will recover from first stimulus and it will be ready for the next one. Right? In this period, okay, so it will get ready for the another impulse. ठीक है जी? Okay. Okay, बच्चे. So when there is the refractory period now, so nerve fiber, it opens the inactivation channels of inactivation sodium channels. So basically that's why the nerve impulse will always travel in one direction, right? It will not come back, right? It will travel in one direction, right? Because when there is the refractory period, what's going to happen, bache? Nerve fiber will recover from first stimulus by opening the inactivation gates of sodium channels. That's how it works. Okay. Yes. Right. So, bache, when the axon is thick, the resistance will be less. So, in the thicker nerve fibers, the nerve impulse transmission is faster, right? Because the resistance will be less. That's it. These are few points related to that. And this is all about this particular topic. Okay. This is all about particular topic. Let's discuss the next part now. Okay, let's talk about the brain now. Right, so in new NCERT only brain part is given, not even the spinal cord, but still I will suggest you to read about the spinal cord, the reflex section, reflex arc. These things are important. Do not miss it. Okay, do not miss it. Okay, so next part is what? Next part is the neuron, brain. Right, so central nervous system is having brain and spinal cord. So when you talk about the brain, it is encephalon. It is encephalon. So you have three parts here. You know it. You have forebrain, midbrain, and hindbrain. What do you have? You have forebrain, midbrain and hindbrain. What do you have here? You have forebrain, midbrain or hindbrain. Right, bache? Right, bache? These are the another names here. Okay, right? So that's what we have. So prosencephalin, mesencephalin, and then you have promencephalin. So how many parts do you have here? 3. Forebrain, midbrain and hindbrain. How many parts do we have here? 3. Forebrain, midbrain and hindbrain. Okay. So we have a trick here. In the forebrain, there will be cache on delivery. There will be cache on delivery. So cache on delivery means C, means cerebrum, or you can call it as cerebral hemisphere. It is not cerebellum. It is cerebrum or cerebral hemisphere, right? O for olfactory lobes. O for olfactory lobes. So in the forebrain, there will be cache on delivery, cerebrum, olfactory lobes, and then you have diencephalon. Then you have diencephalon, right? And what will be delivered? There is a cache on delivery. The T-H-E, the thalamus, H, hypothalamus, E, epithalamus. So yes, these words are given in NCERT. These words are given in NCERT. So when you talk about the forebrain, prosencephalin, cerebrum is there or cerebral hemisphere, right? The largest part of the brain, olfactory lobes, then diencephalon, the dienthalamus, hypothalamus, epithalamus. So thalamus is what? Hypothalamus is what? It is the thermoregulator as well. Now, hindbrain is like, I want to study physics, chemistry and maths, right? And maths. I want to study physics, chemistry and maths. So maths, M stands for medulla oblongata. C here stands for cerebellum. It is the little brain, cerebellum. It is the little brain, cerebellum. And P stands for your pons variouli. These are the parts of hindbrain. So in the paper, they can ask you, what are the parts of forebrain? What are the parts of midbrain? What are the parts of hindbrain? So when you talk about the midbrain, so here you have C2CQ. What's the trick? You have C2CQ. What is the trick? You have C2CQ. Yes. C2CQ. C2 means crura cerebri. And you can also remember crura cerebri. Cruel uncle means cerebral peduncle. The another name here is cerebral peduncle. The another name here is cerebral peduncle. So crura cerebri. So C2CQ. CQ corpora quadrigemina. Corpora quadrigemina. So these are the parts of midbrain. These are the parts of midbrain. Got it? Right. Corpora quadrigemina. Corpora quadrigemina. And the another word here is crura cerebri, cerebral peduncle, midbrain. So C2CQ, so forebrain is having cache on delivery, cerebrum, olfactory lobes, diencephalon, the dienthalamus, hypothalamus, epithalamus. Here C2CQ, crura cerebri, cerebral, corpora quadrigemina. Crura cerebri is also known as cruel uncle, cerebral peduncle. Okay? Then you have midbrain, so it is having pons variouli, cerebellum is there, medulla oblongata is there. Medulla oblongata is there. So that's what you need to keep in your mind. And the midbrain is having these two parts. Okay? Okay. So you know it very well. Right? See, this is the human brain. Look at this. Look at the diagram here. Right? So this part, this convoluted, that is folded part, this convoluted part. Right? This is your cerebrum. This part is the cerebellum. Right? Cerebellum. Look at the pituitary gland, amygdala, brainstem here. So for here, definitely I'm going to suggest you to go for the NCERT. Right? Definitely you have to go for the NCERT. Okay? Okay? Chalo. So now let's discuss it. Look at this wonderful diagram. You know that our brain, very important organ. Very important organ. And let me tell you, it's not like that. The maximum oxygen in our body is used by brain. No, it's liver. it's liver that will use the maximum oxygen, then it is brain. Then it is brain, right? Right, and you know that if brain cells, if they don't even get the oxygen for milliseconds, the stroke can occur, paralysis can occur, right? Brain, very important it is. So look at this. Look at this. You know that we have the skull. We are the craniates. We have the skull here. We have the cranium here, brain box made by the fusion of 8 bones. Right? We have already completed the locomotion and movement. So I hope you people know. So we have the cranium. We have the cranium. We have the brain box that is made by the fusion of 8 bones. Remember? Frontal, parietal, temporal, occipital, ethmoid, sphenoid. Hope you remember. Okay. Now when you look at this, right? Definitely this cranium is there to protect us. And here you have the soft part. Right, here you have the soft part, this convoluted part, we will discuss about it. But now when you focus here, this is your skull. This part is your skull. Here you will talk about further coverings and we call them as meninges. What do we call it? We call it as meninges. Right, what do we call it? We call it as meninges. Is that clear? Is that clear? So brain, the part of our central nervous system, right? It is inside the cranium. Okay, it is inside the cranium. Yes or no? Yes or no? And it contains around 100 billion neurons. I'm not talking about the neuroglial cells. I'm saying it contains around 100 billion neurons. Okay, bache? Right? So if you talk about the brain, it is 1200 to 1400 gram, bache. It is 1200 to 1400 gram, and still we don't feel it. We don't feel heavy here. Why? Because it is in cerebrospinal fluid that will provide the buoyancy, right? That will provide the buoyancy. We'll discuss about it. Okay, we will discuss about it. So now I'm saying that brain may, what do you have? It is covered. It is protected by meninges. It is protected by cranium. And brain is having around 100 billion neurons. So we need to discuss about what? We need to discuss about the meninges, that protective coverings. And for that, the trick is dab. It's not pad, it's dab. It's not pad, it's dab. And not just the human brain, even the spinal cord, right? Even the spinal cord is protected with the meninges. Even the spinal cord is protected with the meninges. Are you getting my point? So what are we talking? We are talking about the brain. What are we talking? We are talking about the brain, right? Brain. And then we are discussing about the meninges that are going to protect it. So the trick here is dab. The trick here is dab. Dura matter, arachnoid, something related to arachnoid, something related to arachnid, spider. I'll come to this, then pyramid. So that will be the sequence from outer side to inside. That will be the sequence from outer side to inside. So they are layers that are protecting our brain, right? 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 Yes. So this one is obviously the outer thick layer of fibrous tissue. This one is obviously the outer thick layer of fibrous tissue. This one is obviously the outer thick layer. Outer thick layer of what? Outer thick layer of fibrous tissue, and that lines the cranial cavity. That lines the cranial cavity. It resists the movement of brain, right? It resists the movement of, yes bache, it resists the movement of brain. Understood? Right? Right? So it resists the movement of brain where? Inside cranium. So it will not let the blood vessel to rupture. Definitely. Right? Right? So the stretching and breaking of blood vessel will not occur. So the stretching and breaking of blood vessels, stretching and breaking of blood vessels doesn't occur. Done. Then you have arachnoids next to it. So bache, it is middle, thin and webby, web-like. It is middle, thin and web-like, webby. Right? And even it is porous. even it is porous, right bache? Right bache? Even it is what? Even it is porous. So yes, do remember this. Let's say if it is the dura mater, this one is the arachnoid. Here you will have a space in between. That space is your subdural cavity. And this cavity, remember, this cavity is having a fluid. Filled with the fluid that is not cerebrospinal fluid. CSF is not there. It is not the cerebrospinal fluid. Right? Here in this cavity, you are not going to see what? You are not going to see the cerebrospinal fluid. Done, bache? Done, bache? Okay? Okay? Right? So next layer is your pia mater. So it is innermost. It is tough, highly vascularized and pigmented. Highly vascularized and pigmented. And pigmented. Understood? So obviously the cavity will be there in between. The cavity will be there in between. This is important. This is important. Yes. This is important. So this cavity is what? This cavity is what? It is your subarachnoid cavity or space. And bache, it is filled with, yes, it is filled with your cerebrospinal fluid, right? It is filled with cerebrospinal fluid, okay? And along with cerebrospinal fluid, some connective tissue strands are also there. Some connective tissue strands are also there. Is that clear? This part clear? Sure? Yes? Yes? So now the next thing is important. Jaise ye dekho. Now you have this. You have the pia mater here. You have the pia mater here. It is fused with brain. Obviously, at places it is fused with the cerebral part. I'm writing the brain only. So it is forming choroid plexus. Choroid, we have anterior and posterior choroid plexus. Choroid plexus. So these choroid plexus, they secrete cerebrospinal fluid. That's a previous year question. That's a PYQ. That's a previous year question. That's a PYQ. Understood? Right? So at places, this pia mater is fused with the roof of the brain, that is the choroid plexus, and that secretes the cerebrospinal fluid. Right? That secretes the cerebrospinal fluid. Okay? Okay? Sure? Yes, please. Sure? Right? Okay. So, overall, you know that brain, the part of central nervous system, which is ectodermal in origin. Right? Ectodermal in origin. So, I told you already. I told you about the parts as well. I told you about the meninges as well, dura mater and the space. Now, the cerebrospinal fluid. What is the cerebrospinal fluid? Yes, question can come from this. You should know about it, bache. Right? You people should know about cerebrospinal fluid. And later on for the brain part, though, you can simply follow the NCERT, right? Right? Whatever is written there, just complete it. Even today, I am not going to teach any extra thing regarding the brain, but yeah, this cerebrospinal fluid is important. And spinal cord is also easy. You know about the spinal nerves, right? And when it comes to the spinal cord, you should know the reflex section, reflex arc, and that is something we study in the class 10th also. ठीक है जी? In the class 10th also. So now we are talking about cerebrospinal fluid. Definitely it is alkaline in nature, right? It is clear alkaline in nature. It is a clear fluid. It's a clear fluid and it is alkaline in nature. And let me tell you, lymph is not there in brain. Here you have cerebrospinal fluid. Here you have cerebrospinal fluid. Okay bache? Okay bache? So this is present in the, yes, the CSF is present in the ventricles. It is present in ventricles. Ventricle means the spaces, the cavities in the brain, right? These are the cavities in the brain. These are the cavities in the brain, right bache? Lined with the ependymal cell. Okay, so CSF is present in ventricles and in subarachnoid space of brain and spinal cord. Obviously your spinal cord needs to be protected. Okay bache? And now when you talk about it, as I said, it is formed by the choroid plexus. It is formed by the choroid plexus. So how that choroid plexus will form? When this pia mater will fuse with the roof of the brain. Okay? Okay? Yes? Done? So you know that what is the function of it? Acts like a shock absorber. Acts like what? Acts like a shock absorber plus maintains buoyancy. And we don't feel the weight of our brain, right? It is around 1200 grams to 1800 grams, right? It is around 1200 grams to 1800 grams, right? Sometimes we say 0.14 kg or 0.18 kg. So obviously we don't feel it because of this CSF. Okay, because of this CSF. So it keeps your CNS moist and maintain constant internal pressure. It keeps CNS moist and maintain constant internal pressure. That's what you need to discuss. That's what you need to know. And it maintains what? It maintains the constant internal pressure. So we are talking about the CSF. We are talking about the CSF. Okay? Done? So, that's the coverings. Ye dekho, look at the diagram. Right? See this. This is your astrocytes. You know that they also support the neurons. Right? Here you have the subarachnoid space that is having your what? That is having your CSF. Right? These are the meninges. And not this one. I have one more diagram, and that diagram is also good. Right? I have uploaded many diagrams, but I just forgot to show you. Ye dekho, look at this. I told you, calcium ions will come when action potential will release here. So these synaptic vesicles, they fuse here. Neurotransmitter will release. They'll attach with the specific receptor on the next neuron. And then again, the membrane polarity will change. Hai na? And see this diagram. Ye dekho, calcium ions, they will enter. Right? The neurotransmitters fuse here. And see this, how the ion channels will move. So diagrams are amazing. We'll share the, right? We'll share the PDF in our official Telegram group as well. Okay, and this diagram is my favorite. It is my favorite. Look at the steps also. Action potential reaches presynaptic terminal. Depolarization of presynaptic terminal will open the calcium ion. Calcium ion will come in. So they will trigger the release of neurotransmitters from vesicle. So neurotransmitter will bind to the receptor sites on the postsynaptic membrane. So opening and closing of channels will cause the changes in postsynaptic membrane potential. So action potential propagates through next cell. Okay? So neurotransmitter will again be inactivated or transported back to presynaptic terminal. That's how it works. Okay? That's how it works. And now the next diagram. Brain. और ये देखो, ये भी meninges. Even your spinal cord is protected with that, na? So you see skin, fat, muscles, these ligaments, this epidural space, then dura mater, arachnoid, right, subarachnoid space with CSF, pia mater, and the spinal cord. Look at the layers here. And these are your vertebral columns, intervertebral discs, right? Right? And ye dekho, this diagram. See, the site of fluid production. The site of fluid production, look at this, right? The skull, this part is the skull here. This is the direction of flow of fluid, right, bache? So cerebrospinal fluid is formed in thin-walled capillaries surrounded by epithelial cells called choroid plexus. They line the ventricle walls. Ventricles are the cavities in between, right? So ye dekho, so they will form here, and here. This is the site of absorption. Okay, so after circulating, cerebrospinal fluid is reabsorbed into blood through these arachnoid granulations, right? These are the projection of that particular meninx. Okay, and look at the cavities here. So skull, cerebellum, this is the, see, this is also showing us the fourth ventricle, the spinal cord. This is the third ventricle. So ventricle word is for the cavities present. This one is fourth, right? Look at this. This one I'm talking about. Okay, this one is fourth and this one is the third. Okay, and these two are the lateral ventricles, right? Lateral ventricles. 1 minute. Ye dekho, your meninges, right? Ye dekho. Dura mater, subdural space is not having the cerebrospinal fluid, right? Then pia mater is there, but center one is this. Okay, this is your cerebral cortex, and this part is cerebellum. Done? Diagrams are good now. And see where your olfactory bulbs are present. Then cavity. Okay, here you have this, right? When you use the word ventricles, you are talking about the cavity. So write a short note on the ventricles of brain. How many ventricles are there? Lateral, third, fourth, right? Do write it. What is cerebral aqueduct? Because it is also given in NCERT. Jaise ye dekho, right? You have that. This is the largest part of our brain. What is it? Cerebrum. Right and left hemisphere is there. Again, I'm repeating, it's a PYQ. Two cerebral hemispheres are connected with a track of nerve fibers, corpus callosum. Corpus callosum is the word. Okay? So now in these hemispheres also, these cavities, lateral ventricles are there. And they are connected with this third ventricle. Look at this. They are connected with this third ventricle, and there is interventricular, means in between these ventricles there is a foramen. Foramen mane opening, right? Jaise, remember foramen of Monro. One is foramen of Magnum. Foramen of Magnum is here. Here it will be foramen of Monro. I'll check it. Yes. And this one is the fourth ventricle. Jaise ye dekho, these are two cerebral hemispheres. The space in between, the cavity in between, you are calling it as lateral ventricle. Even the first, second, you can call it first and second. In between, these two are connected with third ventricle. Then there is a cerebral aqueduct, right? Cerebral aqueduct that is further carrying it to the fourth ventricle. Finally, here in the central canal. Look at the diagram. Right? But it is the interventricular foramen. Done, bache? So this is the cerebral aqueduct. So it's a duct that passes through midbrain. So as I said, it is given in your NCERT. Okay? Look at this. It's a wonderful diagram. One more is there for you. Ye dekho. Lateral ventricles, interventricular foramen, which connects lateral ventricle and third ventricle. It connects lateral and third. So 1, 2, in between third. This is the cerebral aqueduct, right? This is the cerebral aqueduct. Look at this, right? This is your third ventricle. Lateral, third, cerebral aqueduct. So one foramen, one opening is connecting it, right? So cerebral aqueduct connects the third and the fourth ventricle. This information is important. So cerebral aqueduct passes from the midbrain. It connects your third ventricle with the fourth one. Right, bache, with the fourth one, and then there is the central canal. I think this diagram can make it clear. Okay? Yes. Done? Done, bache? Right, so this interventricular foramen, as I said, this one is foramen of Monro. Foramen of Monro. Foramen of Magnum is there in the occipital bone that will connect from where your, this medulla oblongata will further combine with spinal cord. So this is foramen of Monro that is connecting your lateral ventricles with the third one. Right, with the third one. I think I have one more diagram. Baki ye dekho spinal cord. In the spinal cord, the meninges will protect, but here your gray matter will be there in between, forming this S-shaped structure, and your white matter is in the surrounding. But in your brain, your cerebral cortex will have the gray matter, and inside will be the white matter, having myelinated nerve fibers, right? So look at this. So Baki, here you guys can see, I'm just showing it, I'm not explaining it, you have to read it, because spinal cord, spinal arc, the reflex section, reflex arc, you know about it. Okay, even in class 10th we discussed. So look at this part, dorsal root. This is the dorsal root ganglia. You don't have this thing on the ventral side, right? Right? That's how it is. This is the spinal ganglion actually. Okay. Baki, for the brain, as I said, NCERT is important, right? They are not going to ask anything extra from you people. So I believe now cavity part is clear. These ventricle parts are clear. And the question can come from meninges. This is important. The question can come that that cerebrospinal fluid is present in subarachnoid space or not. So you should know that. So look at these convolutions, these enfoldings. This is your cerebral cortex. This is your cerebral medulla in the brain. Okay, okay, bache. It is having gray matter. This one is having white matter, right? This one is having white matter. Chalo, so I'll highlight the important terms here. That's it. So look at the forebrain. So having cerebrum, thalamus, and hypothalamus. And remember my trick. It is COD and D is having THE. Thalamus, hypothalamus, and epithalamus. You have to be specific here. You have to be specific here. So cerebrum forms the major part of the human brain, and it is divided into two longitudinal halves. So bache, right part, right cerebral hemisphere controls our left part, left cerebral hemisphere controls our right part. And it's a cross connection, okay? And moreover, just mostly people, they use the right hand, but left-handed people, they are less, right? They are not that much in number, and they are more creative because if, they are left-handed, means the right part of their brain is controlling it, and it is more concerned with the creativity part. So in our brain, different regions are there, right? But still scientists are working on it. Just say your hippocampus is related with the memory part, right? Your hypothalamus, thermoregulator it is, right? Amygdala is there, okay? Limbic system is there. These are the most frequently asked questions from this particular topic, right? Baki, I told you a deep cleft is dividing the cerebrum longitudinally. So in the paper, instead of longitudinally, they can also use the word transverse. So be clear about it, right? And these are the two right and left cerebral hemisphere. So they are connected by a tract of nerve fiber, corpus callosum. So what is the meaning of tract? Bundle, a tract of nerve fibers. If it is tract, collection of what? Tract word we use for the CNS, na? Tract word we use for the CNS, right? So that's what you need to remember, right? So corpus callosum, important MCQ. Then the layer of cell which covers the cerebral hemisphere is cortex, and it is having the folds, the convolution, sulcus and groove. So cerebral cortex is the gray matter due to its grayish appearance, and neuron cell bodies are concentrated here, giving the color, right? So gray matter is having neuron cell bodies. Cell body, that nuclei are present here. So cerebral cortex contain motor area, you know the meaning of sensory area, and large areas which are neither clearly sensory or motor, they are mixed. So these are the association areas. They are responsible for the complex functions like intersensory association, memory, and communication. So in the paper they can ask you, right, what is the role of association areas? In the statement based question they can ask, right? So fibers of the tract are covered with myelin sheath when it is the white matter, right? Okay. So do write the difference in between gray and white matter. So cerebrum wraps around a structure called thalamus. Again, a previous year question, right? Right. So this part is the major coordinating center for sensory and motor signaling. Next question is related to hypothalamus, bache. You know that it is supermaster gland, right? It controls the pituitary, plus remember it, right? That it controls the body temperature. So it is the thermoregulator. It is also a PYQ. It is also a PYQ. I'm highlighting the PYQs here only, right? So the urge for eating and drinking, right, will be controlled by it. And you know that it is having the neurosecretory cells that will form the neurohormones, right? So that's why it is the supermaster gland. Already we have discussed that in chemical control and coordination. So inner part of cerebral hemisphere, you have the deep structures, amygdala, hippocampus, right? They form the limbic system. So question can come, which parts form the limbic system, right? So along with hypothalamus, this limbic system is involved in the regulation of sexual behavior, expression of emotional reaction. Honestly speaking, brain is a very interesting topic. When we talk about the brain, we talk about the different regions. We even discuss that how, right? We even discuss that how, you know, that different regions, they are associated. Some regions are associated with speech, some with memory, some with comprehension. Like, but obviously, if I'll start it, you'll be like, Q, why, ma'am? What's the need? It is not going to come in NEET examination. So that's why I'm just giving you the overview here, and I'm just highlighting the PYQs here, but do revise it. Baki midbrain is located between thalamus of the forebrain and pons of the hindbrain, and it is having cerebral aqueduct. So this duct bache passes through the midbrain, and you know that it will connect third ventricle with the fourth one. It connects third ventricle with the fourth one, right? So midbrain mein you have that 4 round swellings. So corpora quadrigemina is there. Hindbrain में these 3 parts. So you know that medulla is having a regulatory center for respiration, related to the breathing and all is there. So ये देखो, pons consist of fiber tracts that interconnect different regions. Cerebellum, little brain it is. Little brain it is. So it has a very convoluted, folded space. Arbor vitae, tree of life. It looks like that, right? So in order to provide the additional space. So medulla is connected to spinal cord. They contain centers which control respiration, cardiovascular reflexes, and gastric secretion. And next important question is, right, brain stem is made up of midbrain, pons beroli, and medulla oblongata. That's all. That's all. So brain to just complete from the NCERT, that will be sufficient. So this is all about this chapter, right? You requested me and I took the class. So do revise it. Now ear part is deleted, but again, okay. Again, bache, right? I'll say one thing. That's my suggestion. That do revise the spinal cord, the reflex section thing. Very simple it is, so I'm not going to discuss. I told you about the meninges there, but rest you have to discuss, okay? And don't forget to join our batch. So to join our batch, in the description box of this video, you have the link. Please do not delay. We are discussing cell cycle and cell division. So till January, we have to complete the most important, right? We have to complete the most important chapters of biology, right? We need to target till January only. Your batch will, right, we'll complete your syllabus till March 31st. And in the month of April, bache, there will be 30 full syllabus tests. Every week you are going to get tests. Every week you are going to get tests, right, bache? So part tests are also there, full syllabus test is there. But we have to complete the most important chapters till Jan, right? So that, you know, you can revise it later in February and March, right? So the complete syllabus, we are going to discuss the complete syllabus, that too from scratch. Okay, so do join the batch. We are discussing cell cycle and cell division. Okay, and I'll share the PDF in the Telegram group. And for our batch also, there is a separate Telegram group. So you'll get the link of that group only when you join our batch. Okay, so take care. Bye-bye. If you like the session, do let me know in the comment section. Thank you, everyone. Thank you.